Alright, cool. So I'm gonna go through all the AQA A-level biology topic 1 PMT sheets patterned here for topic 1 biological molecules. So all these sheets here that you see on PMT website here, I'm gonna go through every single sheet. Obviously I can't show the actual sheet, but I'll link it in the description so you can see it. And also like I'll be going through it structurally here. So hopefully it helps you, you can pattern all of that. And yeah man, you'll be setting it. You just gotta do all these PMT sheets and you'll be patterned, bro. Alright, calm. Yeah, I'm gonna go through the first topic 1 sheet, yeah, which is monomers and polymers. Yeah, as always, I can't show it online on the screen, otherwise I'll get striked. So I'll put it in the description. You can go follow that and you can go see the situation. Yeah. Alright, cool. Let's start with this then. Question 1A. It's actually 1C, yeah, according to the thing. So it says, describe the chemical reactions involved in the concentration of conversion of polymers to monomers and monomers to polymers. Yeah, give 2 named examples of polymers and their associated monomers to illustrate your answer. Yeah. Alright, calm. So first of all, yeah, what I would do here is talk about the definition of condensation reaction and definition of hydrolysis here. So write that a condensation reaction joins monomers together, yeah, and forms a chemical bond. Yeah, you always got to talk about the chemical bond. Yeah, that forms a chemical bond. Yeah, and also releases water. Yeah, so it does them 3 things. Yeah, if it joins monomers together. Yeah, forms a chemical bond and also releases water. Yeah, alright, safe. A hydrolysis reaction. Yeah, does the opposite. Yeah, so it's gonna break the chemical bond. Yeah, between monomers. Yeah, because remember these polymers they're made up of monomers. Yeah, which means that the chemical bond is between the monomers. Yeah, and obviously it uses water. Yeah, that's why it's hydrolysis. Yeah, it uses water. Yeah, alright, calm. Now, what you also need to say, yeah, it says give 2 examples here of monomers and their polymers, yeah. So talk about amino acids, yeah. Amino acids obviously undergo condensation reaction to form polypeptides, yeah, with a peptide bond, yeah. Always when you talk about like polymers that are formed, you always gotta mention what type of bond is formed, yeah. So amino acids are the monomer, it's gonna get turned into polypeptides, that's a polymer, but it's forming a peptide bond, yeah. Alright, calm. And then alpha glucose forms starch, obviously, yeah. You can also say it forms glycogen as well, yeah. Under for a condensation reaction, yeah, to form a glycosidic bond, yeah. So again, you got to talk about the bond, yeah. So this is obviously glycosidic, yeah. Alright, calm. Alright, question 2, yeah. So questions two's got like a table, yeah, and you gotta look through which one is which. So question 2A. So the first one, yeah, it says a monomer. Uh Yeah, a monomer in an enzyme's active site, yeah. So obviously enzyme is made out of protein, yeah. The protein monomer is amino acid, yeah. So it's gonna be B. B is amino acid. You got the amine group on the left, you got the acid group, carboxylic acid on the right. That's amine group, yeah. Second one is a monomer in cellulose, yeah. So remember, yeah, that cellulose has got the hydroxyl groups, yeah, on the different sides. So it's got OH on one side, OH on the other side, and H on one side, H on the other side, yeah. Not on the same side. So that means that it's D, yeah. Alright, cool. That's because that's beta glucose, yeah. For that's a monomer in cellulose, yeah. Next one it says is produced during photosynthesis and respiration, yeah. So respiration especially and also photosynthesis, yeah, is gonna form ATP, yeah. So that's molecule C, yeah, that's ATP, yeah, clearly, yeah. Alright, calm. And then the last one, yeah, says forms a polymer that gives a positive result with Biuret test. That's only proteins, yeah. And remember, Biuret detects the peptide bond specifically, yeah. So that's why it's got to be a polymer. It can't just be amino acid. So if the one that forms a polymer that has a peptide bond is obviously B, yeah. So therefore it's the amino acid, yeah. Alright, calm. Again, 2A ends there. And then question 3A, yeah. Question 3A says, what's the definition of a polymer, yeah? So you should know this here easily, yeah. A smaller repeating unit from which larger polymers are made, yeah. Alright, calm. Alright, cool. So that's it for that sheet, yeah. Alright, save. So this is the second sheet here in the topic one topic here, which is called carbohydrates here. Okay, cool. As always, the link will be in the description here to the sheet. Alright, calm. Question 1A, yeah. So we saw this in part one, yeah. You gotta go for each one, yeah. So the first one says is a monomer in an enzyme's active site, yeah. And an active site obviously is made of protein, yeah, because an enzyme is made of protein, yeah. So therefore it's obviously gonna be part B, yeah, because that's an amino acid, yeah. Alright, calm. Next one is a monomer in cellulose. So obviously that means that if it's cellulose, it's gonna be beta glucose, yeah, which is obviously D, yeah, because in beta glucose the hydroxy groups here are on opposite sides and the H are on opposite sides, yeah. Alright, calm. Next one, yeah, it says produced during photosynthesis and respiration, yeah. So that's obviously ATP. You need to produce ATP for respiration and for photosynthesis, yeah. Alright, calm. And then it says forms a polymer that gives a positive test with Biuret test, yeah. So for Biuret, yeah, that's gonna be a protein again. So that's gonna be obviously B because that's gonna be amino acid, yeah. Alright, calm. Alright, question part B, yeah. Okay, cool. So part B, yeah, says that we got a triglyceride, yeah, that's formed from all these three things mixed together, yeah. But what you gotta remember is that after each condensation reaction, you're gonna remove one water, yeah. So they all have the same formula. So if you add together two of the same things, yeah, for now, so C6H12O6, yeah, plus C6H12O6, that's gonna form, yeah, C12, yeah, because you got 6 and 6, that's 12, yeah. So C12, H, but you're gonna remove one water molecule. One water molecule is H2O, yeah, so two hydrogens are removed. So it would have been 24, yeah, but now it's 22. And then O, 6 plus 6 is obviously 12, but obviously you need to remove one oxygen, yeah, because H2O, yeah. So therefore it's gonna be 11, yeah. Alright, calm. Now, obviously add this to the other molecule, since it's triglyceride, yeah, so C12, H12, H22, O11, yeah, and then plus C6H12O6 here. That's gonna get you obviously 4 plus 6 is 18 yeah, so C18 yeah, and then H22 plus another H12. That's gonna get you H32, H34 actually, yeah. But you need to remove 2 because H2O was released here, so obviously H32, and then 11 plus 6 is 17 yeah, but obviously 1 got removed here, so that's gonna be 16 yeah, so O16 you get. Alright, calm. That's the end of that, yeah. Alright, cool. Question C. So question C is asking, yeah, how to do non-reducing sugar test, yeah. So it's only 3 marks. You just gotta mention 3 things. Obviously, to do non-reducing sugar test, yeah, you gotta do a couple things first, yeah. So it's non-reducing, so it didn't get reduced. You need to break it up first into its monosaccharides, yeah. And to do that, you obviously heat with HCl and um sodium hydrocarbonate, yeah. For this one, you don't have to say that. You can say hydrolyze or something like that. You don't have to actually specify HCl and that, yeah. But it's better to do it because sometimes you need to actually say it, yeah. And then obviously heat with Benedict's here. Obviously, we're gonna do the test basically, Benedict's test again, and then a red precipitate will appear here. Since obviously the test was confirmed, so obviously it's gonna break down into the monomers. The monomers are gonna get detected. They're gonna be reduced here. So therefore, a red precipitate will occur here. It will go from blue to red here. Alright, cool. Question 2, yeah. A diagram shows the structure of two alpha glucose molecules, yeah. Draw a box around the ones that connect. So obviously they get connected using, they obviously remove water molecule. So it's bait. So this is what's on the screen here. If you look at it on your sheet, it looks like that. The same shit on that side as well. So obviously the two groups that are gonna react here is this and this, yeah. So just draw a box around each one, yeah. So OH from there will be used with the H there. It's gonna emit H2O and then it's gonna leave a glycosidic bond, which is just that oxygen bonded to the other thing, yeah. Alright, calm. So it will form a bond like that, yeah. Alright, calm. Question B, yeah. Question B says a student carried out a Benedict test here. So just a method other than using a colorimeter that this guy could use to measure the quantity of reducing sugar in a solution. So all you need to do for this, yeah, sometimes I like to ask you little like fugazi kind of questions here. All you need to do here, bro, is just say that filter and dry. Yeah, because obviously you need to filter it, you need to dry it to make sure that there's no excess. The precipitate, obviously, I'm talking about. And then finally, find the weight. So just dry it and then find the weight. That's it. That's the way to quantify it. Yeah, so you don't actually have to use anything to do it. Yeah, calm. Safe. Question C. Yeah. Explain the results for because A and B in a table. So you got A and B, yeah, and it says that one of them is gonna be maltose, yeah, and one of them is gonna be glucose, yeah. So obviously, glucose is a monosaccharide. Maltose is a disaccharide, yeah. So obviously, to do this, it's very simple. If something's red and it stays red, yeah, that means that it just the same amount of it got destroyed, yeah. However, he's gonna add an enzyme, yeah, that breaks up maltose, yeah, called maltase, yeah. And so when maltose breaks up, it's gonna produce more monosaccharides. Do you see what I'm saying? So if it produces more monosaccharides, because it's gonna break up into what's made of glucose, yeah, that's gonna make even more precipitate. So it's gonna be even more dark red, yeah. So that means that B is clearly maltose and A is clearly glucose, yeah. So just write that. So BKA is maltose, is glucose, and B is maltose, yeah, as more. As more precipitate formed, yeah, after maltase action, yeah, you can just speak like that in that kind of way. Talk about like maltase action, yeah. Whenever there's an enzyme, just state the enzyme's name and put action after it, yeah. Alright, calm. Question D, yeah. It's asking you why that's obviously needed. It's obviously because like if it's not using a calorimeter, yeah, you're gonna have to like it's gonna be subjective in it. That one's darker than that. That's not darker than that's obviously subjective. So this makes it quantitative, yeah. So right, it is quantitative, yeah. So it gives you quantitative data, yeah, which means that it's obviously gonna standardize the method, yeah. Always when you talk about quantitative data, yeah, say that it standardizes the method, yeah. That's really what you need to say, yeah. Standardize it because it means that it's gonna be like 100% for anybody to use it, yeah. Alright, cool. E, yeah. So it's talking about percentage uncertainties, yeah. So obviously, the measuring cylinder, he did 15 of that, yeah, and it has an uncertainty of 1, yeah. So what you need to do is you need to just do 1 over 15 times 100, yeah. That's gonna get you 6.67%. And then he used something else for the second one. What did he use? Yeah, he used a graduated syringe, yeah, which has uncertainty of zero point 0.5, and obviously he's using it to measure 5 centimeter, yeah. So 5 centimeter cube of that, so that's 0.5 divided by 5 times 100, yeah, that's just gonna get you 10%, yeah. So obviously now you just add them together. So the total uncertainty is 16.67%, yeah, and that's the final answer. Alright, calm. Alright, next question, yeah, question 3A. Alright, cool. So ask again, yeah, what's the definition of a monomer, yeah? So monomer is just a smaller repeating unit, yeah, from which larger polymers are made. Alright, calm. Safe, bro. Question B, yeah. Lactulose is a disaccharide formed from one molecule of galactose and one molecule of fructose, yeah? Other than being disaccharides, give one similarity and one difference between the structures of lactulose and lactose. So obviously, lactose is galactose and glucose. The other shit is galactose and fructose, yeah? So that's obviously the difference, yeah? The similarity, you can say that they both have glycosidic bonds, yeah? Or you can just say that they both contain galactose, yeah? Which is obvious, yeah? Calm. It literally tells you that, fam. Alright, cool. So both contain galactose here, and then obviously as well, um, lactulose. So the difference here is that lactulose contains fructose here, whereas lactose contains glucose here. So nice, easy, yeah. Alright, calm. Question 4. Question 4a, yeah. Glycogen and cellulose are both carbohydrates. Describe two differences between the structure of cellulose and glycogen, yeah. So you should know this easily from topic 1, yeah. I told you to memorize that. So obviously cellulose is just made from beta glucose, yeah, whereas glycogen is made from alpha glucose, bro. That's one thing. Polymer of beta glucose, whereas glycogen is polymer of alpha glucose. Calm, and obviously cellulose has a straight chain, yeah. Anything with like beta glucose here has straight chain, yeah. So cellulose has a straight chain, whereas glycogen is branched, yeah. Obviously you can talk more, yeah, but that's enough, yeah. But you can talk more as well. You can say that cellulose has 4 glycosidic bonds, whereas my man has 6 glycosidic bonds. You can talk all of that stuff, yeah. Alright, cool. And part B, yeah. So it's talking about what's the beneficiaries of starch here. So two features of starch that make it a good storage molecule. There's a lot of features, so you can say as well that it's insoluble, so it doesn't affect water potential. That's the one that I like to rinse. So insoluble, so it doesn't affect water potential, because this applies to a lot of different molecules, so you can get that instant mark. And then also just say that it's branched, hence it's compact here. So it can be easily stored here. And also, not just that, yeah, but also you can also say that it's branched, so you can have like more surface for the enzyme to act or anything, yeah. It's calm. Alright, cool. It's only two things that I need to do here. Number C, it says obviously state which one. Man said number C, you know. It says tick the box that identifies a test which could be used to show the presence of starch here. So obviously that's iodine, yeah. So that's the last one, yeah. Iodine is always the one, yeah, for starch here. So iodine test here. Alright, calm. Question 5A, yeah. In mammals, in early stages of pregnancy, a developing embryo exchanges substances with its mother via cells in the lining of the uterus. At this stage, there is a high concentration of glycogen in the cells lining in the uterus here. Describe the structure of glycogen, yeah? So you just got to describe glycogen. So obviously glycogen has certain things. Obviously it's a polymer of alpha glucose. Always when something tells you to describe it, just say what it's a polymer of, yeah, straight away. Polymer of alpha glucose here. With a branch structure. That's all they need to get here. Because I think it's only 2 marks, I believe. Yeah, it's only 2 marks. That's all you really need to say. You don't need to describe anything else. During early pregnancy, the glycogen in cells lining in the uterus is an important energy source for the embryo. So just how glycogen acts as an energy source. So anything that acts as an energy source, it just gets broken down into glucose or whatever later on that's used for energy, yeah? So just say that. So part B, just say it is hydrolyzed. That's the proper term for like getting broken down here into glucose. And glucose is used in respiration, you can say. Alright, cool. Safe. Question 6 here. Question 6a. It says that name the monomers here from which a molecule is made of here. Maltose here is made of obviously just glucose and glucose here. So I can just say glucose here. Alright, cool. Part B here. Part B says name the bond that's involved in maltose here. So that obviously that's a form of disaccharides. So obviously it's gonna be glycosidic bonds here. Any sugars they got glycosidic bonds here. Alright, calm. Part C. So in this question, yeah, you got a dilution series, yeah. So I'll make dilution series easy, yeah, man. So you got a table, yes. Obviously, you got concentration of maltose solution, yeah. So for concentration of maltose solution, the way dilute series works here is that you got volume of maltose here, and then you got volume of water, obviously. So therefore, if you're gonna fill in the concentration units here, it's just gonna be mole per dm cube. It's always mole per dm cube for that, no problem. It's always for concentration, yeah? And on the right-hand side, it's asking what is the header for that, yeah? It's obviously going to be volume of water, yeah? Because that's obviously what you're diluting it with, yeah? So volume of water, yeah? And that's going to be in centimeter cube. Okay, safe. Then you also need to find the concentration, yeah? So listen, yeah? For dilution series, yeah? It's simple. If you think about it, yeah? It goes from 0.6 to 0.1, yeah? If I'm doing it in that order, so concentration of motors on the left, volume of motors in the middle, and volume of water on the right, yeah, that means at 0.6, if I want the maximum concentration, it means I've got the maximum amount of motor solution, which it tells you in the question is 15, yeah, and I've got zero amount of water, yeah. From here, you can slowly build up, yeah. So if you think about that, for 0.6, if I want half the concentration, yeah, so 0.3, that's gonna half, so 7.5 and 7.5 is gonna be balanced, yeah, it's gonna split. So I got halfway of water, halfway of that, yeah. Obviously, it's been increasing each time, so 0.6, 0.4, 0. Whatever, yeah, you get calm. So now if I want so now if I want 5 and 10, that means this increased by one increment. It decreased by 2.5, which is one increment. This increased by one increment. So that's just going to be the next step, which is 0.2. You get it. And then you can keep doing it all the way down, yeah? All the way to 0.15. You get it. Yeah? Alright, calm. Same way how here can be 0.5 and 0.4, and that's going to decrease, that's going to increase, yeah? By 2.5 each time. Yeah? If you don't know how to calculate how much it's going to increase by each time, yeah? It's just obvious, it's like on a graph scaling, yeah? Alright, calm. So 0.2 is the concentration for that, yeah? Alright, safe. The question D, yeah. So obviously he's got results on a graph here. So it's explain how you would use the graph to determine the motor's concentration at 0.5, yeah, light absorbance. So what you're gonna do is you're gonna draw a straight line, yeah, line of best fit. Obviously you don't actually need to do it. It's just asking you to describe how to do it. So just say the student will draw a line of best fit, and then my man will read off 0.45 on the concentration on the light absorbance or whatever, and see where that corresponds to concentration of motors here. So just write that into words here. So draw line of best fit, and then read of volume at 0.45, yeah? Alright, cool. Question 7a, yeah? So it's asking explain the difference in structure of starch and cellulose. Yes, that's a common question. Obviously, starch is, there's a lot of differences here, but what I'd write first is that starch is formed from alpha glucose, whereas cellulose is formed from beta glucose here. And then obviously I can say that uh, position of hydrogen. So it says to use the molecule above here. So obviously you know that the position of hydrogen on a beta and alpha glucose are inverted here. So position of hydrogen and hydroxyl group here on carbon atom 1 here. I've explained why it's carbon atom 1 in topic 1 here. Is inverted. Alright, calm. Part B here. Explain one way in which starch molecules are adapted for their function in plant cells here. So you can say a couple things here. So first of all, I would say that starch molecules are obviously insoluble, so it doesn't affect water potential, yeah? There's only one mark here. I mean, two marks here, but it's for just that one thing because it says only explain one way, yeah? So that's it. So it's just insoluble. That's one mark, so it doesn't affect water potential, yeah? Alright, calm. And then part C, yeah, states is asking why cellulose is good, why it's adapted to its function. Yes, obviously cellulose are adapted. Explain how they're adapted. Yeah, so you got to talk about fibrils and all that kind of thing. Yeah, so just say that in simple terms. They are long and straight chain. Wait. are held together by hydrogen bonds here to form fibrils to provide strength here. So obviously they provide strength in the cell wall, and that's what they do here. Alright. So yo. Alright, calm. So this is sheet 3 inside the topic 1 topic here. This is the lipids sheet here. So I'm gonna go through all of that and I'm gonna talk about it. Alright, calm. So question 1, yeah. Alright, cool. So question 1A says that you got name the group represented by COOH here. So obviously the group represented by that is just the carboxylic acid group here. So anything with COOH, that's carboxylic acid here. Alright, calm. Alright, part B here. Part B says shows the structure of a fatty acid, the group R here. Name the type of R group shown in figure 1 and explain your answer here. So the type of R group here, that's the R group, that whole thing here. There's only two types. If it's got a double bond here, that's unsaturated. If it's got no double bonds here, that's saturated. So in this case, obviously unsaturated, yeah. The reason for that is obviously double bond here. Alright, calm fam. Describe how you would test for the presence of lipid in a liquid sample of food, yeah? So this is a standard one, bro, yeah? You should know this by now, yeah? Easy, bro, you get me? C is obviously gonna be add ethanol, yeah? So you're obviously gonna add alcohol first here to make it dissolve in the ethanol, yeah? Because lipid dissolves in ethanol. And then add water. And then shake. And then if it's a positive test, yeah, a white emulsion will appear, yeah? Alright, cool. Alright, calm, fam. Alright, question 2A, yeah? Describe how a triglyceride molecule is formed, yeah? So talk about how a triglyceride molecule is formed. So obviously what's gonna happen here is you got triglyceride, yeah? So you got 3 fatty acids here and 1 glycerol, yeah? So 1 glycerol. So always when it asks you about like how shit is formed, you need to talk about like what's it made of first of all, yeah? And also talk about like condensation reaction and all that, yeah? So and 3 fatty acids, yeah, undergo condensation reactions, yeah? And always when you talk about condensation reactions, yeah, talk about the water that's released, yeah? And 3 molecules of water are released. Obviously, there's 3 molecules of water, yeah, because there's 3 condensation reactions, yeah. And also, you talk about the bonds that are made, yeah, and 3 ester bonds are made, yeah. Obviously, the 3 ester bonds, yeah, are between the fatty acids and the glycerol. So ester bonds are formed here. Alright, safe. Alright, part B, yeah. Table B shows a probability of amino acids, yeah. Put a tick in the correct box. So if you look at the thing, yeah, it will be like a little on the sheet, yeah. In question B, yeah, it will be like 4 different diagrams here. I need to tick the correct box. The correct box is obviously the third box here. Polymeric acid is an unsaturated fatty acid represented by diagram K, yeah. If you look at K, that's obviously unsaturated, yeah, because you got a double bond, so that's correct, yeah. Alright, calm. And obviously, if you look at K inside the pyric acid inside the thing, yeah, it will also be correct in it. It will have 16 carbons or whatever, yeah. Alright, calm. So third one, yeah. Alright, calm, bro. Alright, question 3B, yeah. Because it starts on B, yeah. Alright. Says, describe how an ester bond is formed in a phospholipid molecule, yeah. So obviously a condensation reaction occurs, yeah. Between glycerol and a fatty acid, yeah. That's all you need to talk about, yeah? So you can talk about 2 fatty acids, yeah, is what's made between, but you can just talk about that and that because only 2 marks here. You don't even need to specify that there's 2 fatty acids, yeah? But obviously 2 fatty acids are condensed with the 1 glycerol, yeah? To release 2 molecules of water, yeah? Alright, calm. Question 3B, yeah? My bad, yeah? Question 4A. Alright, cool. So name the 2 parts here. P is obviously the glycerol, and then those 3 Q's here are fatty acids, yeah? So glycerol and fatty acid. Alright, cool. B, yeah. Name the type of bond between P and Q in the diagram. So the bond between P and Q is obviously gonna be that's obviously gonna be thingy, yeah. That's obviously gonna be ester bond, yeah. Alright, calm. Alright, C. Describe how test a lipid sample for the presence of lipid and how you could recognize a positive result. So to test for lipid, yeah, and recognize a positive result, yeah, we should know this by now. Yeah, add ethanol, add water, shake it, white emulsion appears, yeah, standard, bro, you get me? Yeah, add ethanol, then water, then shake, and a white emulsion will appear, yeah. Too easy, bro. They need to like start leveling it up, honestly, you know, they need to just start changing the test, you get me? Like, clock the whole thing, you know what I'm saying? Alright, 5A. Describe how you would test a piece of food, yeah, for the presence of lipid. Bro, they're just repeating questions at this point. Like, come on, man. Add ethanol, yeah. Then add water. Then shake. And then a white emulsion will appear, yeah. Alright, safe. Question B, yeah. Alright, safe. So it says, what's the part labeled A? So if you look at the thing, you got two fatty acids coming out of it on the phospholipid. You got a phosphate head, and you got that part A over there. That part A is obviously glycerol, yeah. Alright, cool. So part A is glycerol, yeah. Glycerol, yeah. Alright, calm. Alright, safe. Alright, cool. Yeah. It's gonna ask what's the bond. I'm gonna predict it, yeah. It's gonna ask what's the bond. Trust it. Yeah, yeah. Part C. You see that? Name the type of bond, yeah, between A and X. So, bro, the bond between glycerol and fatty acid are obviously ester bond, yeah. But you see that, bro? Like, I don't even need to like, bro. At this point, I don't even need to see the paper. Do you know what I'm saying? We don't even need to see the paper anymore, yeah. We just know what he's gonna ask. You know what I'm saying? We know the answer before we even see the question, bro. You get me? That's the levels that we're at here, man. Alright, question D, yeah. Question D says, which of the fatty acids X or Y in the figure above is unsaturated? Well, man, I'm gonna look at which one has the double bond in it, obviously, yeah? So you go up, you look which one has double bond. Oh, I see a nice little double bond on the right-hand side, amino acid. Which one? I mean, fatty acid, bro. Which one is that? That's Y. So therefore, Y, as it has a double bond, yeah? So D is Y, as it has a double bond, yeah? Say nine. Cool. E. The scientists expressed their results as a percentage of lipid in plasma or membrane by mass. Explain how they would find these values, yeah? Bro, so percentage of lipid plasma and membrane by mass, yeah? So they're gonna take the mass of each one, yeah, divided by the total mass, I'm assuming, yeah? So they're dividing the mass of each lipid, yeah, by the total mass and then times it by 100, yeah, to get the percentage of lipid in plasma membrane by mass. You get me? Yeah? Does that make sense? So you're trying to see how much percent of one lipid it is compared to the whole of it. So you're just gonna divide. Alright, safe. That's E, yeah? F is even F, bro. That's the end of the question, end of the sheet. Yo, yo. Alright, cool. This is the fourth sheet in topic one, yeah, called proteins and enzymes, yeah? I'm gonna go through the whole sheet now, yeah? Alright, cool. Question one. So question 1A, yeah? It says name the type of it starts on 1B, yeah? Name the type of enzyme that's gonna hydrolyze the bond labeled G, yeah? Always, yeah? The if it's in between the like amino acids, yeah, that's always endopeptidases, yeah. If it's on the outside, that's always exopeptidases, yeah. Alright, cool. So that's endo, yeah. Alright, cool. Endopeptidase, yeah. C, yeah. C says give the number of different R groups in the polypeptide shown in the diagram above, yeah. So the R group is gonna make each amino acid different, yeah. So just look how many different amino acids there are by seeing how many different shapes there are, yeah. So as you see, some have certain characteristics here. Some have a square shape, some have a circle shape, some have a triangle shape. So there's three types of shapes here. So therefore there's three amino acids you get. Alright, cool. Question D, yeah. A scientist using enzymes to digest whatever calm use information in the table above to calculate the number of polypeptides, 6 amino acids in length and 20 amino acids in length. Alright, cool. So what you want to do here, yeah, is you want to find the total, like the amino acids that are remaining. So as you see, yeah, there's obviously two polypeptides of each length, and there's 5 amino acids in that polypeptide. So 5 times 2. So the total amino acids here was 101. So you want to find the remaining amino acids after those two were used up here. So 101 minus 5 times 2, yeah, because that's the 5 and 2 amino acid, and then 15 and 3. So you're going to take away the 15 times 3 one as well. And that's going to leave you, yeah, with 46. So now, that means it tells us here that we got an amino acid with 6, a polypeptide with 6 amino acids and a polypeptide with 20 amino acids here. So therefore what I'm going to do is I'm going to take this 46 here, and it's going to be 6x here plus 20y, because the number of of polypeptides with 6 is unknown and the number of polypeptides with 20 is unknown. I'm going to call it x and y, yeah, and make that equal to 46. Now just make y equal to 0 first, yeah. You'll see that it doesn't work out. Yeah, it doesn't give you a number for x, yeah, if you worked out. Make y equal to 1, yeah, it doesn't give you a number. Yeah, make y equal to 2. Yeah, make y equal to 2, yeah, and then now we're talking, yeah. Alright, calm. Before you do that, yeah, divide everything by 2 as well, because you can divide everything by 2. So 3x plus 10y, yeah, is equal to 23. So now put 2 into there, you get 3x here, plus 20y, plus 20 is equal to 23. So bring 20 to the other side, you're gonna get yeah, you're gonna get 3, divide 3 by 3, you're gonna get x is equal to 1, yeah. So x is equal to 1, y is equal to 2. So in the table, 1 is gonna go for that one, for the one that's 6, and 2 is gonna go for the one that's y, which is the one with 20. Alright, come. Question 2a, yeah. Describe the induced fit model of an enzyme action and how an enzyme acts as a catalyst, yeah. Alright, cool. So obviously the substrate binds to the thing, yeah. So substrate binds to the active site. Yeah, that's what's gonna happen. The substrate is gonna bind to the active site, yeah. And the active site is gonna induce a shape here. It's gonna change its shape here. Change its shape to accommodate, to obviously accommodate the substrate here. So that is complementary here. Complementary means it's got the same shape as the substrate, yeah. Come to the substrate. Alright, save. That obviously reduces the activation energy, yeah. Alright, calm, because it puts strains on the bonds here, so it reduces activation energy, yeah. But no more time, you don't have to quote that, yeah. If it's only 3 marks, you don't need to quote that. Alright, calm. Scientists investigated the action of enzyme ATP synthase, yeah. They made reaction mixes containing that and that, yeah. Tick one box to show the other substrate that scientists must add to the reaction mixes to produce ATP, yeah. So obviously ATP is made with PI, which is inorganic phosphate, and also ADP, yeah, adenosine diphosphate, yeah. So it's just the second one, yeah. So B is the second box, yeah. So second, yeah. Alright, go on. Alright, cool. C, yeah. C says suggest and explain a procedure the scientists could have used to stop each reaction. So here we go, an enzyme reaction. To stop an enzyme reaction, you need to denature the enzyme, yeah. The easiest way, yeah, is bro, what's going on there, bro? C, yeah. Alright, calm. The easiest way, yeah, is to obviously just boil it, yeah. Boil to denature ATP synthase, yeah. But you can also say other things as well. Like you can say, you know, put like an acid in there, put whatever, put bare substrate, put whatever. I don't know, yeah. It's calm. Alright, cool. Yeah, D, yeah. Explain the change in ATP concentration with increase in inorganic phosphate concentration here. So obviously, as you see the graph here, if you increase the inorganic phosphate, you're gonna have the ATP produced is gonna increase, obviously, yeah. But then it reaches the top where it reaches a plateau. So you need to talk about that. So just say as inorganic phosphate, PI, yeah, concentration increases, yeah, more enzyme, substrate, complexes form, yeah. Listen, you need to start talking like this here. You need to start talking like enzyme substrate complexes form here. You need to start using words like that. They like that. That's what they're looking for. That's the key words, you get me, that are gonna get you the marks quickly. Do you know what I'm saying? So that's the whole point, yeah. So obviously, if you add more PI, the enzyme that's making the PI ATP synthase is gonna react faster. It's gonna make obviously increase like more enzyme substrate things, yeah. Until 40 mole per dm cubed, yeah, which if you look on the graph, that's where the plateau is hit, yeah. 40 mole per dm cubed, where all active sites are occupied, yeah. So the reason obviously why it plateaus here is because all there's no more the enzyme becomes a limiting factor, yeah. You need more enzyme now to deal with all the substrate that you put in there, yeah. All the active sites have substrate inside, so it's finished, yeah. Calm. Question 3A. Student investigated starch hydrolysis using enzyme, calm, whatever. It's not even 3A, right? No, it's draw a table and the student could use to record all his results, yeah. So obviously the table that he's using, he's gonna use starch concentration, which is that's the independent variable, yeah. And then the dependent is just how long it takes for hydrolysis here. So just obviously write that in the table. So starch concentration, yeah. In these situations, when it asks you for a graph or a table or something, just think what's the independent variable, what's the dependent variable, and that's it. Starch concentration, yeah, is gives it to you in milligrams per decimeter cube, yeah. Okay. And then the next column is going to be time for hydrolysis here in seconds, yeah. You know what? Put hydrolysis 1, yeah? Because it says that he repeated the experiment, yeah? So what you can do is you can just write here hydrolysis 2, hydrolysis 3 if you want, yeah? Time for hydrolysis 2 seconds. Right, come on, bro. Alright, safe, yeah? Alright, whatever, bro, yeah? Alright, come on. Yeah, bro, I'm actually dumb, man. Alright, cool, cool. Alright, question B, yeah? Alright, cool. Question B. Describe the results you would expect the student to obtain. So obviously, as starch concentration increases, the time taken to hydrolyze starch increases, yeah? Obviously, no shit, yeah? Cool. Obviously, got more starch here, so obviously it's going to increase the time it takes to destroy the starch here. Calm. C, yeah? A competitive inhibitor decreases the rate of an enzyme control reaction, yeah? Cool. So explain how, yeah? So competitive inhibitor obviously takes the active site, and therefore nothing else can bind to it. So you need to talk about that, yeah? Again, use certain type of language, yeah? So say the inhibitor has a similar shape to the substrate, yeah? That makes it obviously complementary, yeah? And binds to the active site, yeah? Cool. Hence prevents formation of enzyme substrate complexes, yeah? Okay, cool. When you talk about like that, yeah, they'll give you that 3 marks easily laid out. Do you see that, bro? You get it? So like say about the inhibitor is complementary, yeah? Say about that binds to the active site, blocks it, yeah? And always say something like hence prevents formation of enzyme substrate complexes. They like when you say that kind of thing, yeah? Alright, calm. Alright, question D, yeah? Assuming the scientists are correct, so just how eating stale bread could help to reduce weight gain, yeah? So it says when bread becomes stale, the structure of some of the starches change. This change starch is called retrograded starch, yeah? Scientists have suggested retrograded starch as a competitive inhibitor of amylase in the small intestine. So you need to just talk about how So why is that a good thing? Obviously, it means that if it's an inhibitor, yeah, less hydrolysis of starch, yeah, hydrolysis of starch here to maltose, and hence less absorption here of glucose here. So obviously, starch is going to be hydrolyzed by the enzyme here to maltose here, but unfortunately, since the enzyme has blocked it, since it's been blocked here, it can't do that. So obviously, it can't go down to maltose, which means that it can't be absorbed as glucose here. Safe. Question for you. Okay, cool. Describe how the structure of a protein depends on the amino acids it contains. Yeah, so bro, if you see like a 5 marker talking about structure of protein, yeah, it means that you need to talk through every single structure. And you need to talk primary, secondary. That's easy marks. Yeah, the primary structure. So just literally just stay it out for them. Yeah, the primary structure is the sequence and order, obviously. Yeah, order of amino acids here come in the chain. Secondary structure is formed by hydrogen bonds. And obviously, tertiary structure. You can also talk about alpha helix and beta pleated sheets there as well. Is formed by ionic bonds and disulfide bonds here. Normally, when you talk about the tertiary structure, you don't need to say both of them. Yeah, you can just label one of them. You can just say ionic bonds or disulfide or hydrogen. Doesn't really matter. Alright, come. Come. You always need to say the tertiary. Yeah, the tertiary is what's responsible for the active site. Yeah, it's what's responsible for the unique shape in the active site. So you can state that there for another mark. Come. Quaternary structure. Yeah, contains more than one polypeptide. Obviously. Yeah. So that's just the standard definitions for all of those, yeah? Alright, cool. Question 5, yeah? Okay, cool. Question 5e, yeah? Describe how an amino acid joins to form a polypeptide. So there is always NH2 at one end and COOH at the other end, yeah? So obviously, yeah, one amine group here joins to a carboxyl group, yeah? That's how it works, yeah? So obviously, yeah, how this forms is the peptide bond here is formed between the one amine group, yeah, and one carboxyl group, yeah? So therefore, on the other ends here, you're still going to have one amine group for free and one carboxyl group that's also free, yeah? That's why it's like that. Alright, cool. Question 6a, yeah? Cool. Explain how the active site of an enzyme causes a higher rate of reaction. Yeah, so you need to be careful about the question here. So it's asking you, yeah, explain how it causes a higher rate of reaction. Yeah, so obviously just say that induced fit. Yeah, causes the active site to change shape. Yeah, come on. So they want to know how actually like lowers activation energy, yeah? So that's why you got to state that obviously lowers activation energy. That's why it's faster reaction, yeah? When you talk about why that happens, obviously it happens because there's stress on the bonds, yeah? The reason why there's stress is because obviously induced fit, yeah? Come. Question B. Other than those stated, yeah, give one factor that the student could have controlled in the investigation. So obviously he could have controlled the mass of the potato, yeah? Alright, cool. The student gave the maximum rate of reaction a value of one arbitrary unit. So, alright, cool. So if you look at the com, so if you look at the table, yeah, the maximum is 1 unit, yeah. So that means that the maximum is 6 here, and the thing. So 6 is 1 unit. So calm. So 6 is 1 unit, yeah. So 6 corresponds to 1 unit, yeah. Alright, calm. So now think about it, yeah. If 6 is 1 unit, then how much is 18? The time of 18. It means that you're gonna have to basically decrease it in a way, yeah. So therefore, yeah, think about this. The rate of reaction is increasing, yeah. It should, if it's a lower time, so if it's 6 seconds, yeah, it should be a faster rate of reaction compared to 18. So all the numbers above now, so like 18, 10, 7, yeah, they should all have rate of reactions that are below 1. You get it? 1 is the maximum, bro. They should all be below 1. So what you gotta do here is obviously you gotta do like 6 divided by each one, yeah. So 6 divided by 18, 6 divided by 10, 6 divided by 7, yeah, to get obviously the ratio, yeah. So when you do that, you'll get a table like this. You'll get 0.33, 0.60, 0.86, and then 1 and 1. You get it? So 6 to 1, 6 to 1, and then that will be to each one ratio independently, yeah. So you do 6 divided by each time, yeah, to find the rate of reaction, yeah. Cool. Alright, so if question D, yeah. So basically, for question D, you need to plot the graph here. So this is the data that you were using here, and then here you got the hydrogen peroxide concentration. So just write mole per dm cube, and then rate of reaction here, arbitrary unit, yeah. Alright, cool. Go up in just like 0.5, yeah, all the way up to 3, yeah. Since the x-axis is going up in that anyway, yeah. Alright, cool. And then for the y-axis here, just go up in like 0.2, it's minor, yeah. That will cover most of it, yeah. And so now just plot basically 0.5 there, 1 there, 1.5 there, and so etc, yeah. And then eventually what you want to do now is you want to connect it, yeah. For this one, you need to connect it line by line. So make sure you connect it like one by one, you get me? So one by one, yeah, not line of best fit, yeah. And then what you can do as well is you can extend it down to 0 as well if you want, yeah. Alright, cool. Alright, question E, yeah. It says that suggest a change that the student could make to his procedure so that 10 centimeter cube of oxygen, yeah, would be produced in less than 6 seconds, yeah. So obviously increase the surface area of the potato, yeah. If there's more surface area, if you cut it up, it's gonna increase the rate of reaction, yeah. Cool. Question 7, yeah. Describe the biochemical test to confirm the presence of protein in a solution, yeah. So obviously add biuret, yeah. So add biuret reagent. Obviously it turns purple, yeah, if it's positive, yeah. Come B. Dipeptide contains of two amino acids joined by a peptide bond. Dipeptides may differ in the type of amino acids they contain. Describe two other ways in which all dipeptides are similar and one way which they might differ, yeah. So the way they're similar, yeah, they all have one amino group. They all have an amine group here at one end. And another way they're similar is you can say they all have a carboxylic group here. So that's calm. That's a bit of a cheap mark here, but you can also say that they all contain carbon, nitrogen, hydrogen, and oxygen, yeah. Calm. And then the difference here Yeah, differences, they contain different R groups here. Obviously, that's what makes the amino acids different, yeah? Calm. C. Explain what the positions of the spots in the diagram show about these amino acids, yeah? So you drop them there and they move towards a negative electrode, yeah? If they're moving towards a negative electrode, they're obviously positive, yeah? So you can say that they move to the negative electrode because they're positive, yeah? Yeah, cool. So, however, the spots are gonna have a different distances, yeah, that they travel due to the different charge to mass ratio. Yeah, you can say different differences in charge, yeah, but when you talk about stuff like charge and that kind of thing moving across, like like time of flight and that kind of thing, yeah, talk about charge to mass ratio, yeah. Calm. Which makes sense in it. Depending on the mass here and the charge, it's gonna move differently, yeah. Calm. And obviously, one spot has two amino acids. That's why he put three, and there's only two spots because one spot has two amino acids. Yeah, one spot has two amino acids. Yeah, because obviously they have the same charge. Yeah. Calm. Question 8A. Yeah. Okay, cool. Describe how a non-competitive inhibitor can reduce the rate of reaction. Yeah. So obviously, it attaches, yeah, to the enzyme at the allosteric site. Yeah. So that's just a site that's other than the active site, yeah? So like from the back of the enzyme, yeah? Which obviously changes the tertiary structure of the enzyme, yeah? So the enzyme becomes permanently deformed, yeah? And then obviously, so the enzyme is no longer complementary to the substrate. So no substrate can bind, yeah? Or you can say so no enzyme substrate complex is formed, yeah? Alright, cool. Alright, cool. B, yeah? The scientists concluded that pectin is a non-competitive inhibitor of the lipase enzyme here. Use figure 1 to explain why the scientists concluded that pectin is a non-competitive inhibitor, yeah? So if you look at when there's pectin, yeah, if you add lipid concentration, yeah, what should happen if it's competitive, yeah, it will kick it outside. It will if you add, if it's even if it's blocked here, if you add more substrate, it will kick out the thing that's blocking it, yeah. But because non-competitive like permanently deforms it, yeah, it doesn't matter if you add more substrate, if you add more shit, it's still gonna remain the same. So that's what you need to write. You need to write that high substrate concentration, as you see in the graph. When you add more substrate, it doesn't change shit. It's still plateau, bro. It's still like flat gradient, yeah. High substrate concentration doesn't overcome inhibition, yeah. Come. Question 9A, yeah. Cool. Describe how a peptide bond is formed between two amino acids to form a dipeptide, yeah? It's 2 marks here, so you just gotta say that condensation reaction, yeah? Between amine, yeah? And the carboxyl group here. So between the amine and the carboxyl group, there's obviously a condensation reaction which causes the peptide bond to form, yeah? Calm, B, yeah? The secondary structure of a polypeptide is produced by bonds between amino acids, yeah? Calm, fam. So explain to, yeah, describe how. So secondary structure is produced by bonds. So obviously that's the hydrogen bonds and that kind of thing, yeah? So hydrogen bonds, yeah? And it's obviously between the NH group, yeah, and the double bond O group, yeah? Because obviously C double on O, yeah, is going to be electron negative on the O minus, and H is going to be H plus, yeah? So that's going to create um hydrogen bonds, yeah? To form beta pleated sheets, yeah? And alpha helix, yeah? So C, yeah? So C's asking, two proteins have the same number and type of amino acids but different tertiary structures. Explain why. So they got the same number and type here, but different tertiary structures. So it's obviously because the position was different from the get-go. So it means that the primary structure is different. So they have different primary structure, yeah? They have different primary structure, yeah? And hence, yeah, when it comes to the secondary structure, yeah, the bonds are going to form in different places, you get me? So hence form ionic, disulfide, and hydrogen. Again, you only got to mention one, yeah, but you can just put all of it anyway. Hydrogen bonds in different places, yeah. Come. Question 10A, yeah. Formation of an enzyme substrate complex increases the rate of reaction. Explain why. So, bro, 2 marks here, easy, yeah. It reduces the activation energy, yeah, due to stress on the bonds. That's it, bro. Alright, come. Question B, yeah. Calculate by how many times the rate of reaction is greater with the enzyme present, yeah. So with the enzyme present, 578 will release per second, yeah. Without the enzyme, 3 times 10 to the power minus 9 will release. So you just need to divide the bigger number by the smaller number to find out how many times bigger it was. So 578 over 3 times 10 to the power minus 9, yeah. That's going to get you 1.93 times 10 to the power of 11, yeah. That's man, alright, calm. Question C, yeah. Calculate the rate of reaction of the enzyme activity with no lactose at 2.5, yeah. So look at 2.5. On the bottom of the graph. If you look at 2.5 and go up here, on the y-axis for C, that's about 0.44, yeah? So therefore, I can say that 0.44 is there. And then it's over the maximum, yeah? So calculate the rate of reaction for the enzyme of no lactose, so C, yeah? As a percentage of the maximum rate shown with lactose, yeah? So with lactose, that's L. If you look at the maximum with L, that's 1.4 on the y-axis, yeah? So it's just going to be 0.44 divided by 1.4, which is the maximum, yeah? Times by 100 to make it a percentage, yeah? So that over that to make times 100, 31.4%, yeah? Alright, safe. Alright, come. Question D, yeah? Lactose binds to the enzyme, yeah? Suggest the reason for the difference in results shown in the graph with and without lactose, yeah? So obviously, bro, that's bait, yeah? Obviously, lactose binds to the enzyme, so the binding of the enzyme, the binding altered, yeah? The tertiary structure of the enzyme, yeah? Which caused the active site to change shape. Hence, obviously, because the rate of reaction increased here, it made it in a way that it somehow made it in a way that's not an inhibitor. It makes more enzyme substrate complexes here, so hence more enzyme substrate complexes form here. Calm. Question 11, yeah. You know that because the rate of reaction was increased here with lactose here. Calm. Alright, cool. Something like that. Alright, cool. Question D, yeah. Nah. Question 11A, yeah. Draw the general structure of an amino acid, yeah. So obviously an amino acid, yeah, has obviously it's got C. It's got the carboxylic acid group here, the COOH, yeah. It's got the amine group over there, so H2N, yeah. And then up here it's got R group here, the variable region, and then it's just got H down here, yeah. Alright, calm. Question B, yeah, the genetic code is described as degenerate, yeah. What's meant by that? So that's in topic 4, yeah, you learn about that. Degenerate means that more than one codon, yeah, codes for a single amino acid, yeah. So more than one codon codes for one amino acid, yeah. So what that means here is that you got a codon here is a triplet, yeah, of bases, yeah, that codes for an amino acid, yeah, and more than one can code for the same shit. Alright, cool. You need to actually state one from the example. So for example, if you look at the table that they provide you, go up, look at the table. How it works is the first base you read it from the left here, second base you read it from the middle, and bro, and then read the right hand side as well, yeah. So for example, UCU, yeah, if you look at U on the left hand side, C on the middle, and then U on the right hand side, that's serine, yeah. And if you look at U in the middle, C in the middle, and C on the right hand side, that's also serine. You get. So UCU and UCC is both serine, yeah. So therefore, I can say that. So therefore, for example, UCU and UCC both code for the same shit. Alright, cool. If you don't understand that, go look at the topic 4 video, yeah, and you'll get. Alright, calm. Question C, yeah. What's the minimum number of bases in the gene coding for this polypeptide, yeah? So the polypeptide has those three amino acids, so just add it, bro. So 203 plus 279 plus 300, yeah. That's 782, yeah. And obviously there's 3 in each one, yeah. My bad. The enzyme is polypeptide 465 amino acids long, yeah. So just take 465, that's the amount of amino acids, yeah, and just obviously times that by 3. You get it? Because each amino acid has 3 bases, yeah, for a codon, yes. That's 1395. Alright, cool. Question D, yeah. Use the information from table 1 to tick one box that shows a single base substitution made in DNA that would result in a change from VAL to ALA, yeah, at amino acid number 203. So remember, yeah, that this is RNA inside the base. This is DNA here, but it's RNA inside the actual table, yeah. So remember that because this is DNA, it literally can't be option 2 and option 3 because they got uracil, and uracil isn't even present in DNA. So let's look at the first one, CAA to CGA. In terms of RNA, that's gonna be G, you see? So let's go look up. This is gonna be GUU, yeah? And if you look at the table, it's correct. Look, GUU is VAL, yeah? And then it turns into GU. No, my bad. GCU, yeah? Which would correspond, yeah, to CGA in DNA. So therefore it's the first box, yeah? You get that box 1, yeah? Because that's going from GUU to whatever to GCU, yeah, which is the same as going from wherever to wherever, yeah, from going from CAA to CGA, yeah, because you gotta swap it for RNA. You get. Alright, calm. Question E, yeah. A change from GUL to lyase at amino acid it has 300 had no effect on the rate of reaction catalyzed, whatever. Use all the information in your knowledge of protein structure to suggest the reasons for the differences. So if there's a change at one thing and there's no change at the other thing, yeah, that means that obviously one changed the structure, one didn't change the structure. That's the only difference, yeah. So that means that change at 300, yeah. Otherwise, there's no reason for it to change, yeah. So change at 300, yeah, did not affect active site. However, yeah, change at 200, yeah. It's not even 200, it's 279, yeah. At 279, may have changed the active site. We can say hence here. We also talk about how it changed the active site, yeah? Obviously by changing hydrogen bonds here. That's the main factor here. Alright, cool. And so how is that going to be affected here? If you look at the situation, if you look at the amino acid that it turned into here at 279, GLU turns to LYS here. GLU is what? If you go up to the table, it says that GLU, if you look at the color code at the bottom here, it says positively charged and negatively charged here. So obviously it became positively charged here. LYS is a positive charge here. Calm. So LYS here is positively charged, so therefore I can say that it turned positive here. Obviously it was negative before because it was doing that. Calm. Safe. So for both of them, both amino acids are changed into the same shit. It went both, or you don't even need to say both. You can just say that negatively charged. Positively charged here. Calm. Cool. This is the fifth sheet here inside topic one. This is our nucleic acids here. Calm. Question 1A. Name the type of bond between complementary base pairs here. So obviously between AT and CG, it's gonna be hydrogen bonds here. So question 1A, right, hydrogen bonds here. Alright, safe. And the adjacent nucleotides in a DNA strand here. So obviously adjacent nucleotides, they're held together by phosphodiester bonds here, which makes the phosphate backbone here. So phosphodiester bond here. Alright, cool. Alright, question B here. Alright, question B here. It says that the length of a gene is described as a number of nucleotide bases it contains. Use information in the above diagram to calculate the length of a gene containing 4.38 calm. So look at the diagram here. Between 1.7 nanometers within that enclosed area here, you got 1, 2, 3, 4, 5 here. So you got 5 different lines, 5 different bonds here, which means 5 base pairs within that here. So if it's got 5 for 1.7 nanometers here, that means that you got to find how many for 4.38 times 10 to the power 3 base pairs here. So for 5 base pairs, we've got 1.7 nanometers here. So how many for 4.38 times 10 to the power 3 here? For that many, you need to do that divided by 5 here and then times it by 1.7 here. Obviously, the number here is going to increase here. So that's going to go 1489.2 here. So that's the amount of nanometers here for that here. Alright, calm. Question 2A here. Okay, cool. 2A has got a diagram. And it's asking you to fill in the diagram here according to what goes in there. Yeah? So obviously, if you look at the diagram here, these are all going to be biological molecules here, which means that we're going to have carbon and oxygen here. Alright, calm. So galactose here is just a sugar, so obviously it's going to have carbon here. Then the second one, nitrogen, obviously it's got no nitrogen, it's a sugar here. Oxygen it's got obviously, and then phosphorus obviously not here. Alright, calm. Second one is phospholipid here. So phospholipid is going to have carbon. It's not going to have nitrogen. It's going to have oxygen, of course, because a lipid here. And then it's going to have phosphorus as well, because obviously phospholipid here. Calm. RNA obviously has carbon here. It's got nitrogen as well, because obviously nucleic acid and that. Oxygen, of course. And then it's going to have phosphorus as well, because the phosphate backbone, you get me? Alright, calm. And then sucrose here. Sucrose is another sugar, so it's just going to be the same as the first one here. Alright, calm. Question B here. Ooh. So in question B, there's a little graph here. It's a bit fugazi here, this graph, because it's not even done evenly yet, and it's done in a weird way, of course. But it's minor. So what you want to do here is you want to take, says size of population at time plus 4 hours here. So go to the graph here, go up here. You see on a base it says time. Go to where it says 4 here. Look up from 4. Keep going until it meets the graph here. It's going to meet my man up there, yeah. Now, because it's a logarithmic graph here, it means that you count the base. So like, for example, if that's standard here, that's 1, 2. It's not 2, it's 3. You get me? You count this as the first one. So the first line, second line, third line, not 1, 2. So even if it's 2 boxes above here, it's not 2, it's 3, yeah? You get it? So same shit here. So look for where that attaches to my man. And if you look here, it will be how many boxes above, bro? It will be 2 boxes above here, but because you count the baseline as well, it's going to be 1, 2, 3. And now look at the y-axis here, it's going to be 10 to the power of 9, yeah? So therefore, that reading is 3 times 10 to the power of 9 for 4 hours, yeah? You get it? Alright, calm. Now, look at where it says negative 4 hours, because you need to do divided by negative 4 hours, yeah? So negative 4, look back to where negative 4 is, yeah? Now look up from there, so negative 4. How many boxes above? It's 1, 2, 3 boxes, yeah? But obviously, you got to count the base as well, so that's 4 boxes, yeah? Look at the y-axis, it's 10 to the power of 6, yeah? So it's 4 times 10 to the power of 6, yeah? So 4 times 10 to the power of 6. Alright, calm. Obviously, put that in the equation, yeah? That's going to be log 10 of that. Alright, cool. And then divided by log 10 of 2, yeah? Alright, safe. If you put that in the calculator, it's going to give you 9.5507, yeah? Alright, safe. So now you got that answer. So the number of generations here, if you think about it, yeah, from -4 to 4 hours, yeah, that's 8 hours in total. Because -4 to 0 is 4, and then another 4 is 8, yeah. So therefore, it took 8 hours to get that many generations, yeah. So therefore, if you want one generation, you want to find out the time for one generation, then you need to divide it. So put in a ratio, yeah. So for 8 hours, yeah, it gave you 9.5507 generations, yeah. You want to find how much for one, yeah. So do 1 divided by my man, and then times that by 8. So 1 divided by this, whatever that gives you, do a and s times 8. That's going to get you 0.837, yeah, which makes sense, yeah. If 8 hours took that, then 0.837 hours, yeah, is for 1, yeah. Alright, calm. Question C, yeah. Cool. So question C, yeah, is asking you, which is the correct model, yes? Obviously, the correct model is going to be Q, so just write that. So Q is the correct model. That's semi-conservative replication, yeah. So obviously Q is semi-conservative replication because obviously my man's semi-conservative. You've got one DNA strand that's being left over and then it's being used as a template. Yeah, so let me just explain to you the situation here. So what's going on, bro, is my man used nitrogen 15, carbon 15, yeah, as whatever. Alright. And then he moved it into the he basically put that in there, nitrogen 15, yeah, for a long time. And he moved it into a nitrogen 14 environment, which is obviously lighter, yeah. So obviously, because of semi-conservative replication, yeah, 115 strand, yeah. So think about this, 215 strands, yeah. 115 strand is going to get used as a base. Another 15 strand is going to get used as a template as well. Now, when it makes a new strand, yeah, that's going to be 14 because it's in a 14 environment, yeah. So that's 14 there as well. And now when it replicates again, 15 gets used as a base. 14 also gets used as a base. 14 adds because it's in the 14 environment. Another 14 adds because it's in 14 environment. You get me? Same thing here, yeah. So 15, 14, yeah, 14, 14, yeah. Alright, calm. So that's what's going on here, basically. So that's why there's that number of peaks and whatnot inside the diagram, yeah. So Q is obviously correct, semi-conservative replication. It can't be P, yeah, because if you think about it, if it was P, there would be more than there would be 2 peaks in generation 1, yeah. So it can't be P. Because there'll be 2 peaks in generation 1, yeah. What that means is if you look at P, yeah, in generation 1, you got 2 heavy and 1 and 2 light. If you got 1 heavy strand, 1 light strand, it's not going to be 1 peak. It's going to be 2 different peaks. 1 heavy peak, 1 light peak, yeah. So it's going to be 2 peaks, yeah. So it can't be that. And if you look at R, yeah, it can't be R because it's going to be bare peaks in generation 3. If you look at generation 3, it's got a lot of different shit going on. It's got one with a line, one with an X, one with another line. There's just bare shit, which means that generation 2 and generation 3, it's not gonna it's gonna be more than just 2 peaks, yeah. So just write that down as well. So it can't be R. Alright, safe. Alright, cool. Question 3A, yeah. So question 3A says describe the structure of DNA, yeah. So obviously DNA is what? DNA is a polymer, yeah. It's a polymer of what? Polymer of nucleotides, yeah. Come, so it's a polymer of nucleotides held together by what? Held together by phosphodiester bonds, yeah. which are formed obviously by condensation reactions, yeah? To form, you got to talk about what shape it makes, yeah? It's going to make a double helix shape here, so to form a double helix here, which is held together by hydrogen bonds, yeah? So just think of all the bonds in DNA, you only got hydrogen and phosphodiester, yeah? So think where the phosphodiester are and talk about where the hydrogen is, yeah? So hydrogen bonds here, which occurs between obviously adenine, thymine, and then cytosine, guanine, yeah? Put the and there to indicate that adenine, thymine bonds together and cytosine, guanine bonds together. Obviously, AT is 2 bonds, CG is 3 bonds, but that's not needed for the question, yeah? Alright, calm. Then obviously write as well, each nucleotide, because you need to talk about the structure. So you said it's a polymer of nucleotides. You need to say what a nucleotide is now, yeah? So a nucleotide has a deoxyribose, because that's important for DNA. That's why it's not RNA, yeah? Because it's deoxyribose, yeah, not ribose, yeah? Phosphate and a nitrogenous base, yeah? Alright, calm, bro. That's it, yeah? Alright, save. Question 4A, yeah? Describe how phosphodiester bond is formed between two nucleotides within a DNA molecule, yeah? Alright, cool. Phosphodiester bond is going to be formed in a condensation reaction, obviously, between phosphate and deoxyribose, yeah? So the phosphate and the deoxyribose from the second one form a phosphodiester bond, yeah? And this is going to be catalyzed by DNA polymerase, yeah? So condensation reaction. Which is forming DNA, yes, as DNA polymerase, yeah? And it's going to be between deoxyribose and phosphate, yeah? That's phosphodiester bond, yeah? Alright, calm. Question B, yeah? Okay, so question B, you got some next equation, yeah? And it's asking you to use information and your understanding of DNA structure to calculate the maximum number of amino acids coded by the gene, yeah? Alright, save. So what you need to do here, yeah, is you're going to need to use that equation to find my man. Now, it tells you that there's 168 guanine, yeah? If there's 168 guanine, that means there's 168 cytosine, yeah? Because it's the same shit, yeah? So that means that G is equal to 4, A plus T, minus C, yeah? Bring C to the other side, you got G plus C is equal to 4A plus T, yeah? Now, that means that you got 168 plus 168 is equal to 4A plus T, yeah? By the way, the reason why they're both 168, yeah, is because guanine bonds to cytosine, yeah? So if there's one of guanine, then there's one cytosine. If there's two guanine, two cytosine, you get it. Alright, calm. Now divide by 4, yeah? So add this together, divide by 4, you're going to get A plus T, yeah? Is going to be equal to 84, yeah? Alright, calm. Now, these also have to be the same, yeah? So it's just 84 divided by 2, which means that A is going to be equal 42 and T is going to equal 42. Alright, safe. Now what you want to do is add everything together, yeah? So 168 plus 168 plus 42 plus 42, yeah? That's going to get you 420, yeah? Now, you need to do 420 divided by 2, yeah? Because that's for one strand, yeah? So what you're doing here is obviously each one is a duplicate, yeah? Because there's two bases in each one. But obviously in an actual strand, there's only half of the amount of bases, yeah? So divide by 2 to get how many actual bases you got, yeah? That are coding for something. How many like coding bases, yeah? Which is 210. Now, to make a gene, yeah, you need a codon, yeah? You need a triplet. So a triplet needs to be coded for, yeah? So to find out how many codons, you got to divide by 3 because there's 3 amino acids, yeah? 3 bases per codon, yeah? So divide by 3, you get 70. So that's the amount. That's the final amount, yeah? Alright, calm. Alright, cool. Question C, yeah? Explain the thing associated with protein. It's obviously protein associated with DNA. It's obviously histone, yeah? That's bit. Alright, calm. D. In the process of semi-conservative replication, the two strands within a DNA molecule are separated. Each then acts as a template for the formation of a new complementary strand, yeah? Describe how the separation of strands occurs. Obviously, DNA helicase here is going to break that apart. So it's going to chop that. So DNA helicase here breaks hydrogen bonds between complementary. You gotta use that word, yeah. You gotta talk about complementary, yeah? Between complementary base pairs, yeah? Alright, come. Alright, question 5A, yeah? Describe the role of DNA polymerase in semi-conservative replication, yeah? So obviously polymerase, yeah, is going to make the DNA in it. So it's going to join adjacent nucleotides. So say that it joins adjacent nucleotides, yeah? So whenever you talk about DNA polymerase, yeah, always use the word adjacent nucleotides, yeah? So it's the nucleotides that are next to each other like that, that is going to join them, you get me, to form the backbone, yeah? Whereas this one is the hydrogen bonds between it, yeah? Alright, calm. Joins adjacent nucleotides, yeah? By condensation reaction, yeah? To form phosphodiester bonds, yeah? So always talk about the bonds that's formed, talk about condensation reactions, yeah? Alright, calm, and talk about where it's occurring, yeah? Alright, cool. B. There's no B even, just question 6. Alright, cool. Question 6, yeah? Question 6 says, name 2 scientists who proposed models of the chemical structure of DNA. So obviously Watson and Crick here. That's bit Watson and Crick here. Alright, calm. Question B. It says, name the enzyme used in the DNA replication, yeah? So a scientist replicated DNA to do he mixed enzyme with identical. So obviously the enzyme that's used in this DNA replication to make that is going to be DNA polymerase, yeah? It's about making DNA, yeah, not just breaking it, yeah? So that's why it's not helicase, yeah? Alright, calm. C. Use your knowledge of the semi-conservative replication of DNA to suggest the role of a single strand, yeah? So obviously the single strand is what gets broken up in the first place, yeah? And that's going to act as a template for the free nucleotides to come in, yeah? So acts as a template. Acts as a template. And determines the order of bases. Alright, cool. So obviously acts as a template and it determines the order of bases, yeah? Because obviously the template has the original ones, yeah, which can act as like tells it which ones are formed, yeah? And the second one, the role of the DNA nucleotides, they need to form complementary base pairs, yeah? So they need to form complementary pairs, yeah? That's why they're there, yeah? Alright, calm. Question 7a, yeah? Draw the structure of a single nucleotide, yeah? So in a nucleotide, you got a couple of situations, yeah? You got phosphate head like that. You got deoxyribose here, which is a pentyl sugar here, so therefore it's gonna be like a pentagon shape here. And then you got the base, which is normally a square, yeah, rectangle here. Calm. So phosphate here is up here. You got deoxyribose. And then you got the nitrogenous base here. Calm. Question B, yeah. Give 2 features of DNA and explain how each one is important in semi-conservative replication of DNA, yeah? So 2 features of DNA, yeah? So first of all, it's got weak hydrogen bonds between the strands, yeah? Which allows it to unzip easy, yeah? So write that down. So the weak hydrogen bonds here, because obviously hydrogen bonds are intermolecular force here, is weak here, between bases allow strands to unzip here or unwind here. Obviously by DNA helicase here. And then the second one is obviously that 2 strands here means both can act as a template. Yeah, imagine if it was 1 strand here, you just have one acting as a template, forming the next thing. You get me? It'd be long, yeah? So you got 2 strands here that means that both can act as a template, yeah? Alright, calm. Part C, yeah? There's a lot of shit you could have said there, yeah? But that's just 2, like that. Alright, calm. C, yeah? Replication of mitochondria, alright, calm. So the DNA replication of that only starts after that, so... Alright, so after 2/3 here, that's when it starts here. So find how long for 2/3 here. So 2/3... Calm. So 2/3 here of how many bases in total? There's 16500 bases here. So 2/3 of that here is 2 over 3 times 16500, yeah? That's gonna get you 11,000, yeah? So that's how many bases he's completed in that time. Now, what's the rate of doing it? The rate is 50 nucleotides per second, yeah? So if you're trying to find, so if I've got nucleotides, I'm trying to find seconds here. Think about it, yeah? If I've got the unit here is nucleotide, and this is nucleotide per second, yeah? So if I divide nucleotide by nucleotide per second, yeah, it's gonna flip this fraction, it's just gonna give you seconds, yeah? So therefore, yeah, what you need to do is you need to do 1100, yeah, and then divide that by the rate, yeah? That's gonna tell you the time that it took. So by 50, yeah? So that tells you the time that it took. That's gonna be 220 seconds, yeah? Alright, calm. Alright, safe, yeah? Then that's when it's gonna start the other strand, yeah? So the second strand is gonna start now. So that's at 16500. It's just gonna start at this rate, yeah? So that's all the all the nucleotides, yeah? Not part of it, yeah? So all the whole thing, yeah? Divided by 50, the rate again, that's gonna be 330 seconds, yeah? So now you just add the together 220, yeah, plus 330, that's gonna be 550, yeah? That's gonna tell you the time it took for the whole thing, yeah? So that got done. Then it started the second one. The second one isn't split, it's just the full 16500 divided by 50, you get that. Alright, calm. So that's it, 550 seconds, yeah? So C, yeah? Alright, calm. Question 8A, yeah? Describe the role of two named enzymes in the process of semi-conservative replication of DNA, yeah? So if you want to do that, you gotta talk about two enzymes, yeah? So that's obviously DNA helicase, DNA polymerase, yeah? So let's talk about that, yeah? So say DNA helicase here causes hydrogen bonds to break, yeah? DNA polymerase joins adjacent nucleotides with phosphodiester bonds, yeah? Calm, that's the two enzymes they want there, yeah? Calm. Question B, yeah? Some long question. Cool. Suggest explanations for the results in the table, yeah? Okay, cool. So if you look at treatment D, so treatment D's got the least, treatment E has got the least, and C, the control, has got the most, and then F, you also got the, cool. What's F? F is added RNA that prevents the translation of silin A and added silin protein. Okay, cool, cool, cool. So D is fucking up shit. E is also doing the same shit, and then F is doing it, but it's also gonna add silin as well. So that's why it's higher, yeah? So what you can say here is that in treatment D, silin A, yeah? Silin A, whatever you say, yeah, cannot bind to DNA, obviously, yeah? Calm. Whereas in E, because that's what it literally tells you, antibody that binds specifically to silin A, yeah? So therefore, if it's binding to silin A, it means that silin A can't bind to DNA, yeah? Calm. In treatment E, RNA is obviously going to be interfering, yeah, with mRNA, yeah? If you don't know about this, it's calm, it's in topic 4, yeah? So basically, what's going on here, yeah? In treatment D, yeah, silin A can't bind to DNA in the first place because it's binding to the antibody, yeah? In E, RNA is going to prevent translation of silin A. So if it prevents translation, that's obviously a problem with mRNA, messenger RNA. So if it's preventing mRNA from moving, it means that the silin A can't actually form, yeah, in the first place, yeah, because it's formed from translation and transcription, yeah? And then, because obviously it's a protein, yeah? And then in F, yeah, you're gonna add silin A after as well. So it prevents silin A formation, but you add silin A. So because it has so because it's preventing of added silin A, yeah, it means that silin A can also bond to the thing, yeah. So it can bind to DNA, yeah. Alright, calm. Safe. Question 10A, yeah. No. What question are we on? Question 9, yeah? Question 9a. How many nucleotides are shown above, yeah? So question 9a. How many nucleotides? You got 1, 2, 3, 4, bro. Times that by 2, you got 8 in total, yeah? Alright, calm. Alright, B. Name the bond between the adjacent nucleotide, yeah? Obviously, the bond between the adjacent ones is gonna be um it's gonna be phosphodiester, yeah? Calm. Question C. The enzyme, DNA helicase, and DNA polymerase are involved in DNA replication, yeah? So explain each one. So DNA helicase obviously breaks the bonds, breaks hydrogen bonds between bases. And then the second thing, yeah, DNA polymerase here is gonna join adjacent nucleotides, yeah? Alright, safe. Alright, question D, yeah? Adenine ATP is a nucleotide derivative, yeah? Contrast the structure of ATP and a nucleotide. So a nucleotide, obviously ATP has a ribose and DNA has deoxyribose here. So write that down. So ATP has ribose here, whereas DNA has deoxyribose here. And then secondly, yeah, ATP obviously has triphosphate here, has three phosphate groups, you can say, yeah. Whereas DNA has a one phosphate group here. Alright, calm. Alright, question 10a, yeah. Figure 1 shows a base of DNA of a DNA molecule, yeah? Name the part F of each structure, yeah? So part F is obviously deoxyribose, yeah? That's the sugar, yeah? Alright, calm. Deoxyribose, yeah? Alright, safe. Question B, yeah? Scientists found that a sample contained 18% adenine. Obviously, 50% of it is the same shit. So adenine and thymine are the same shit. So percentage of thymine is going to be 18. And what you can do is just 18 plus just 50 minus 18. So then you just get 32. That's the second one, yeah? Because obviously it would have been 18 plus 18 plus 32. You can do 100 minus 18, then divide by 2. Or you can just get straight to the end here. So that's guanine is obviously 32 because it's going to make up the rest of it, yeah? Calm. C. Name the enzyme shown in part in figure 2, yeah? So in figure 2, it's obviously moving that way and that way. So it's obviously making shit. So that's obviously DNA polymerase, yeah? It's bait because it's moving away from it as you see, yeah? That means it's forming it, yeah? Alright, calm. The arrows in figure 2 show the directions of each new DNA strand is being produced. Use figure 1 and figure 2 and your knowledge of enzyme action to explain why the arrows point in opposite directions, yeah? So obviously the reason for that. Yeah, calm. So the reason for that, yeah, is because DNA has anti-parallel strands here. So what that means is that because it's got anti-parallel strands here, the nucleotides are aligned differently from each one, yeah? That's going to ensure that enzymes here have active sites with a specific shape here. Hence, only substrates with complementary shape can bind with the active site here. It's not really going to ensure it. I don't know why I wrote ensure here. Calm. So, because DNA has anti-parallel strands here, I was asking why does it need to point opposite directions here? So obviously because it has anti-parallel strands, it means that the nucleotides are going to be aligned differently. So therefore, the enzyme's going to move in a certain way that's aligned with that nucleotide, and the enzyme's going to move the other way, which is aligned with that nucleotide. Yeah. Alright, calm. Alright, question 11. Yeah. Draw a box around a single nucleotide. Yeah. So just look at the nucleotides. It's going to look like that. Something like that. Yeah. So then just draw a box around that. Yeah. Whatever. Alright, calm. Alright, question B. Yeah. Complete the table by adding the missing values. Yeah. So it starts off with 16. So that means that there's going to be an 21 here, 34 there, yeah? So that means there's going to be, if there's 16 adenine here, that means that there's 16 thymine in strand 2. And then what you want to do here is, if there's 21 cytosine in strand 1, there's obviously 21 there. And if there's 34 there, there's obviously 34 in strand 1, yeah? Calm. Now just add it together, so just do 16 plus 24 plus 21 here, and take away from, as percentage here, so take away from 100. And you're going to get 29. So therefore, this is just 29, and that's just 29, yeah? Okay, safe. Alright, cool. Question 5C, yeah? Why am I saying 5C? 11C, yeah? Alright, calm. So it says that use figure in part A and your knowledge of enzyme action and DNA replication to explain why new nucleotides can only be added in a 5' to 3' direction, yeah? So obviously, DNA polymerase has a specific shape here. So therefore, it's only complementary to the 5' end here. So if you look at the so obviously the end of the 5' and 3' are different, yeah. And obviously DNA polymerase only complementary to one of them. If you look at the diagram, it's complementary to the 5' one, yeah. That's why it's moving that way, yeah. Alright, calm. Alright, calm. This is the sixth sheet inside topic 1, yeah. ATP, yeah. Alright, calm. Question 1A. Describe how an ATP molecule is formed from its component molecules, yeah. So obviously it's made of 3 components, yeah. Adenine, ribose, and 3 phosphate molecules, yeah. Alright, calm. You gotta talk about how it gets made, yeah. So it's formed through a condensation reaction, catalyzed by ATP synthase, yeah. So ATP synthase is the one that's making that, yeah. Alright, calm. Alright, question 2A, yeah. 2B even, yeah. Name 2 products of ATP hydrolysis, yeah. So you're breaking ATP back into what it's made of, yeah. So it's made of obviously ADP and then inorganic phosphate, yeah. So that's obviously adenine diphosphate, yeah, and then inorganic phosphate. Alright, calm. C. So other than those given, name 2 variables the student could have controlled, yeah. So always, bro, always in biology, yeah. When you talk about control, just think of 2 things immediately: temperature and pH, yeah. And in this case, you got ATP solution. So just say temperature of ATP solution, yeah. And then pH of ATP solution, yeah. Calm. D, yeah. Describe and explain the pattern shown by the data in the table, yeah. So obviously the data in the table is saying that if you got more ATP, yeah, the length of muscle decreases. Obviously, that's because you got contraction occurring. Yes, if you got more ATP, you got more AT available to be hydrolyzed to make energy. Yeah, and that's gonna mean that the muscle is gonna contract and decrease in length. Yeah, so just write that down. So that's gonna be as concentration of ATP increases, length of muscle decreases, yeah? So just state the obvious first here. And then say more ATP hydrolyzed, yeah? So more energy released, yeah? So more muscle contraction, yeah? Alright, calm. Let's save question E, yeah? Hydrolysis of one, whatever is made, that's added, my man. Calculate the energy available from ATP for contraction of muscle in this side, yeah? So we got 30,500, yeah? And how much is it using? 6% of energy released is released as heat. So 40% is the actual useful one, yeah? So that means that you're going to do 30500 times 0.4, yeah? That's going to get you 12200 joules, yeah? Alright, calm. Now, obviously, it says that it's going to use 0.05 centimeter cube. So do that into decimeter, yeah? In decimeter, that's 5 times 10 to the minus 5 decimeter, yeah? Alright, calm. Now, obviously, divide that by 1,000, you get that. Alright, calm. So obviously now you need to look at, for D, yeah? D's got concentration, yeah, is going to be 8. If you look at the table and go up here, it says that D has 8, and then the heading is times 10 to the power minus 6 here. So 8 times 10 to the power minus 6 concentration, yeah? So if D has 8 times 10 to the minus 6 here, and you're trying to find the amount of moles or whatever, yeah? Then you're gonna need to times that by my man, obviously, yeah? You get, so 5 times 10 to the power of minus 5, yeah? Because that's more per decimeter, yeah? Alright, calm. Times that by 8 times 10 to the power of minus 6, yeah? That's gonna get 4 times 10, yeah? To the power of minus 10, yeah? Alright, save. So if that's how many I've got here in the situation, yeah, and it's 122 energy, yeah, released here per each thing, yeah, for one mole per decimeter, yeah, then I'm gonna need to obviously times that by 5 times 10 to the power of, I mean by this, yeah, so 4 times 10 minus 10, yeah, times 122, yeah, 00, yeah, that's gonna get 4.88 times 10 to the power minus 6 joules, yeah. Alright, cool. Alright, question 3A, yeah. Needs to tick in the appropriate box for ATP synthase here. So ATP synthase making ADP plus PI is going to make ATP plus H2O, yeah? So it's the third one, yeah? So ADP plus PI makes ATP, yeah? Plus H2O, yeah? Alright, cool. Obviously condensation reaction, yeah? So it's going to leave water, yeah? Alright, calm. Question B, yeah? The new antibiotic is safe in humans because it does not inhibit the ATP synthase found in human cells, yeah? I suggest why human ATP synthase is not inhibited, but obviously it's because they got different tertiary structure, yeah? So ATP, yeah? If it ever talks about why something gets like inhibited and something doesn't, yeah, it's because different structure, yeah. So ATP from human, yeah. ATP synthase even, yeah. You get the point, yeah. I don't need to write. Alright, cool. Synthase, yeah, has a different tertiary structure, yeah, to bacterial ATP synthase, yeah. Alright, cool. I ain't dyslexic, by the way. It's just I just be tweaking it. Alright, calm. Question 4A, yeah, because I know the spelling is atrocious, isn't it? Alright, cool. Question 4A, yeah. 4B even, bro. I should be tweaking, innit? ATP is an energy source here used in many cell processes. Yeah, give two ways in which ATP is a suitable energy source for cells to use. So obviously, it releases energy simultaneously. So it releases energy instantaneously, not simultaneously. Yes, instantaneously. Yeah, I can't be asked. Alright, cool. And obviously, it can be rapidly resynthesized. Yeah. Alright, cool. Alright, question B. Yeah, not even question B. Question 5A. Describe how ATP is resynthesized, yeah? So obviously from ADP and PI, yeah? So from ADP and inorganic phosphate during respiration, yeah? So that's when ATP is remade, yeah? Catalyzed by ATP synthase, yeah? Come on. And then B, the two ways which hydrolysis of ATP is used in cells, yeah? So it doesn't just say for energy, it says which ways is it used, yeah? So you can say that to add phosphate to substances, yeah? And make them more reactive, yeah? So what it does is it phosphorylates other things here, other molecules here, which makes them more reactive here by changing their shape. Calm. And also to provide, that's the second use here, is to provide energy for reactions here. Calm. That's it. Calm. This is the water part of the sheet here, so the water sheet here. Cool. Question 1A, yeah. Explain 5 properties that make water important for organisms, yeah. So you need to explain 5 properties here, yeah. So you should know these properties here. They're easy memorizable, yeah. First one is that it's a metabolite, yeah. Cool. A metabolite. And hence used in condensation and hydrolysis here. So that's what a metabolite means. A metabolite means something that can be used in condensation and hydrolysis. You know how water is obviously released during condensation, it's obviously used during hydrolysis, therefore it's a metabolite, yeah. Alright, cool. Second thing, yeah, that I can say is that it's a solvent, yeah. So reactions can occur inside of it, yeah. Solvents or reactions can occur, or I can also say that it's a solvent, so therefore it can also transport shit, yeah. Alright, cool. Third thing, yeah, is that it has a high specific heat capacity, yeah. So it has high specific heat capacity, yeah. These are just 3 marks, bro. You get me? These are standard, bro. Yeah. High specific heat capacity. That means that basically, it can use a lot of water. Bro, what am I talking about? It can use a lot of heat, yeah, without increasing temperature. So therefore, it can increase in heat, yeah. It can take in all that heat without actually increasing in terms of Celsius, yeah. So therefore, high specific heat capacity. So buffers change in temperature. So it's gonna buffer the change in temperature. It's not gonna move the temperature that much, but it's gonna change in terms of heat. You get it? So it can store a lot of energy without changing the temperature. You get me? Alright, cool. Then you can say large latent heat of vaporization, yeah? So a large latent heat of vaporization, that's basically gonna provide a cooling effect here. What that means here is that my man is not gonna water, yeah, is not gonna evaporate, yeah, into vaporization, yeah, until it has a certain amount of heat. So it needs to store a lot of heat for that to happen, bro. It has a very large latent heat, yeah. So what that means, if something had a low latent heat, a little bit of heat, it would be evaporating, bro, like ethanol, yeah. But this shit here, it needs a lot of heat to be able to reach them levels, do you know what I'm saying? So therefore, when it does evaporate, yeah, it's gonna move a lot of heat with it, bro. You get me? When you sweat, yeah, it's gonna make you cool down, yeah, because it's gonna release a lot of heat with it, because it took a lot of heat to get water to that stage where it can evaporate. You get me? So yeah. Alright, cool. Cohesion, yeah, as well. So cohesion and adhesion, you just gotta mention one of them. So you can say cohesion. So aids in mass transport, yeah, of the xylem. Or you can say adhesion as well, does the same shit. Or you can also talk about surface tension as well. Shit like that, yeah. Alright, cool. And that's everything, bro. Alright, calm. Question 2C, yeah. Let's see if question 2C. State and explain the property of water that can help to prevent temperature increases in the cell, yeah. So that's going to be a high specific heat capacity, yeah. It's going to help buffer temperature, yeah. So high specific heat capacity, yeah. Obviously, yeah. And it's going to obviously buffer changes in temperature. Alright, cool. Question 3, yeah. Question 3 is basically the same as what we just done, yeah. So explain 5 properties that make it important for organisms, yeah. So question 3A. But just do it again so that it sticks with you. Alright, cool. So just say again a metabolite in condensation and hydrolysis, yeah. Right. A solvent. So reactions can occur. right? High specific heat capacity, so buffers temperature, yeah, right? High latent heat or vaporization, so provides cooling effects. And then finally, right, cohesion or adhesion, yeah, in the xylem, yeah. Alright, calm. Question 4A, yeah. Cool. So right that it's saying which one, yeah, buffers temperature, yeah. So obviously has a high specific heat capacity. So can gain a lot of energy or heat, yeah, without changing temperature. Alright, cool. Alright, question 5, yeah. The two properties of water that are important in cytoplasm of cells, yeah. For each property of water, explain its importance in cytoplasm. So you can say a lot of things here, yeah. So I would say that. So two things that water can do, yeah, is it's a solvent, obviously. So reactions can occur faster, yeah. And then second of all, yeah, as well, is you can also say that obviously it's you can say that it's a metabolite, yeah, that's reactive, yeah, so it takes place in condensation and hydrolysis. You can also say that it's polar. So it acts as a universal solvent, yeah. Water is obviously polar, yeah, which means that shit can diffuse into it and shit like that, other polar molecules, yeah. So you can just give any of those two things, yeah, say whatever you want to say, yeah. Alright, calm. Question 6 is the essay, so I'm gonna talk to you about the essay now, yeah. Cool. So this is the last question, yeah. It says write an essay on ways in which water and the regulation of water content are important to organisms, yeah. So what you wanna do, yeah, is wanna write a little you wanna write a little plan, yeah, so that like anything that's missing from there, they can like check it and it shows that it's structured, yeah. So just think about things here that are in water across the course, yeah. So the first thing here is that obviously water as a metabolite, yeah, and then think about what that means, yeah. So hydrolysis, yeah, and condensation, and then ATP hydrolysis as well. So that's one thing that you can talk about, yeah. War as a metabolite, yeah. That could be a whole chapter, yeah. Now, second thing you could talk about, yeah, is energy storage, yeah. So what things consistent, yeah, like constitute energy storage, yeah. Obviously, glycogen, yeah. Obviously, glycogen hydrolysis, yeah. That's gonna be inside energy storage, yeah. It's gonna use water to obviously hydrolyze glycogen into glucose and that, yeah. And then again, glucose metabolism, yeah. Alright, cool. So that's the two things there, yeah. Alright, cool. Safe, yeah. So that's that, yeah. Now another thing to talk about, yeah, is photosynthesis, yeah. So that's from a completely different part of the course, yeah. So photosynthesis, yeah. So obviously got the LDR, yeah, which obviously contains water, yeah. Then you also got photolysis, yeah. And then you're also gonna have like ATP and NADPH production, yeah. So all these things here involve water, yeah. Alright, come. So that's three paragraphs here, right there, yeah. Calm. Remember, they gotta be free from three different parts of the courses, yeah. So these two are from the same part of the course, it's calm, yeah. This one's from a different part of the course, yeah. So it's minor. Alright, cool. Mass transport, yeah. That's from a different part of the course as well. So that's three different parts of the course there, yeah. Mass transport, yeah. So this is cohesion, adhesion kind of thing. Remember as well, yeah, it should be 4 paragraphs here, and then a fourth one that is extra information, yeah. And another thing as well is you should aim to do like 6 paragraphs here to make sure that's patterned, yeah. Cool. Cohesion, adhesion, yeah. And then transpiration. Then also like about if you think about water inside the mass transport, you can talk about like tug pressure and that, yeah. Calm. Another thing is translocation, yeah. So translocation, yeah, I can talk about water pressure flow. You can also talk about like osmosis inside the xylem and phloem, yeah. So this kind of thing you do in the exam, yeah. You just write like a quick plan, yeah, of all the different topics you can think of, yeah, that contain water, yeah, and then you build it from there, yeah. Another thing is osmoregulation, yeah. So that's another topic completely as well. So I can talk about hypothalamus and that, and ADH and all that shit, and then also blood water potential, yeah. Calm. Another thing as well, yeah, which is covered in I think topic 2 year cells, yeah, is tissue fluid as well. So I can start talking about tissue fluid, yeah, about the um hydrostatic and osmotic pressure, yeah. Calm. And then finally, yeah, not even finally, but another thing, yeah, is I can talk about gas exchange, yeah. So like water across the grill, yeah. Alright, safe. And then lastly, yeah, I can talk about thermal regulation as well. So like specific higher latent heat, yeah, which obviously can help with like cooling effect and that, yeah. So you got bare different topics here, yeah. You can pick 3 from any of these, yeah, and you can create a long essay, yeah. Alright, calm, safe. Alright, cool. So I'm gonna start with the whole essay here, yeah. This is paragraph 1, yes. Obviously I've written the whole essay, and obviously it's a lot more detailed than you need to put in your exam, yeah. And there's bare things going on, yeah. And there's like 8 paragraphs here, but the whole idea is that it's a long essay that you can take whatever parts you want from, yeah. There's obviously a 25-mark essay, yeah. It's worth the marks because bare shit in here, yeah. But you'll see the process of how to do it, yeah. Alright, come. So for this paragraph here, this paragraph 1, yeah, I'm gonna break it down, yeah. So the topics that it contains here is, as we wrote, metabolite, yeah, was one of the first things that we thought of, yeah. So we're gonna make a paragraph based on metabolite, yeah. Now, obviously, we got ATP, which is 316. The second number is a topic here, so it's topic 1, yeah. Water is 317, so second is topic 1, and then proteins and nucleic acid, yes, that's also topic 1. So it's all topic 1 in this chapter, yeah, which means that in the other paragraphs, I'm gonna need to use other topics here. Alright, cool. Remember, they can be from different chapter. They can be from different chapters and still not be marked. Yeah, so it needs to be from different topics. Yeah, not just different chapters. Yeah. Alright, calm. So calm. So let's start here. Yeah? So water is vital to all living organisms, yeah, functioning both as a metabolite and as a medium facilitating, yeah, vital biological processes, yeah. One significant role of water is in hydrolysis reactions, where it's used to break covalent bonds within larger molecules, yeah. So what you're saying there is you're establishing hydrolysis here. Now you need to talk about hydrolysis and condensation and what they do and how they're used, yeah. So hydrolysis is obviously A-level standard. Remember, yeah, you can't use any GCSE information in the essay, yeah. A common misconception here is that the essay is marked as like a marked like on top of each other, yeah. So like if you mention my man, you get this. If you mention that, you get this. Nah, bro. You need to show consistency, yeah. There needs to be extra information scattered throughout the whole thing. There needs to be a paragraph extra information. You need to be talking at like an A-level standard throughout the whole thing. That's how you get 25 marks. You get me? If you're not talking, if you like use a little bit of GCSE and that, yeah, even if you have like a good amount of A-level shit, they're not gonna give you 25 marks. You get me? To have that bracket, it's ranked per bracket. You get me? They read the whole essay and they see what bracket does it fit into. So you need to talk at A-level standard for the whole thing. Yeah, you understand? So here, yeah, a prime example is hydrolysis. So hydrolysis of adenosine triphosphate ATP into adenosine diphosphate and inorganic phosphate. Now, this is a very simple fact here. We all know this here, but look at the way that it's structured here. Look at the way that I formed it here. It says a prime example is the hydrolysis. So we're talking about hydrolysis. We're talking about an example of hydrolysis here. A reaction which is catalyzed by the ATP hydrolase enzyme. Calm. This exergenic reaction releases energy here due to lower Gibbs free energy. So what I'm doing there is I'm using A-level chemistry knowledge here to fill in as the extra knowledge for A-level biology. You see what I'm saying? So instead of just like memorizing extra knowledge for the whole of biology, which you can do in a minute as well. I'll tell you how to do that, yeah. You can use the chemistry knowledge, yeah, to add into that, yeah. So what I'm saying here as exogenic here is that it's going to be a feasible reaction. It's going to happen spontaneously. If you know about chemistry, you know about feasibility and that, yeah, enthalpy change and entropy, yeah, and leading to Gibbs free energy, yeah. So just sprinkle that in, yeah, adds a little bit of flavor to it, yeah. So reaction releases energy due to lower Gibbs free energy of all products, yeah, compared to the reactants, providing energy necessary for various cellular activities, yeah, including muscle contraction. So you want to mention a couple of things, yeah, muscle contraction, active transport, yeah, biosynthesis, so making polymers and that kind of thing, yeah. And then additionally, what is essential in condensation reactions where it's released here during the formation of complex molecules like proteins and nucleic acids, yeah. So again, you're talking about polymers and polymerization, yeah. These reactions are thermodynamically unfavorable, yeah, but they become feasible through coupling, yeah. So that's a little bit of extra information there. You got something called ATP coupling, yeah, which is where the ATP can make the reaction feasible and lower the Gibbs free energy, yeah, with ATP hydrolysis showing water's critical role in both anabolic and catabolic pathways that contribute to metabolism, yeah. Alright, calm. So here, there's not really much being said. We're just talking about hydrolysis and condensation. But look at the way that it's formed. Look at the way that's constructed in a nice way with that extra information. That's what they're looking for. That's a concrete paragraph right there, yeah. You don't need bare shit. That's a concrete paragraph, yeah. Alright, calm. Let's move on to paragraph. Alright, cool. So I got similar shit here. I got paragraph 2 here. So paragraph 2 here, it's got 3.1.2 carbohydrates here, 3.1 water, and 3.6. So got topic 1, topic 1, and topic 6 here. So therefore, I'm also bringing in topic 6 now, which is where I'm going to get the other marks as well, yeah, which is control of blood glucose concentration, yeah. Alright, calm. So furthermore, yeah, so this is paragraph 2, yeah. Water's role in hydrolysis and condensation reactions is fundamental to the metabolism of energy storage molecules. Glycogen, the primary storage form of glucose in animals, is broken down into glucose monomers through hydrolysis reactions where energy is needed, yeah. Excess glucose is stored as glycogen via condensation reactions which release water molecules. These reversible processes are vital for maintaining blood glucose levels and providing energy during periods of increased demand, yeah. So listen, this paragraph you might think is a bit small, yeah, but it's structured in a nice way that it's a nice little bridge. It's a nice little extra thing to put after the first paragraph, you get me? And it shows that without water, yeah, because it's about the importance of water, yeah. So without water, you're not going to have that glycogen being stored because you can't do hydrolysis and condensation, which means that the blood glucose level is not going to be maintained here, which touches on topic 6 a little bit, yeah. So that's what it's about. It's not about mentioning everything, yeah. It's about mentioning the right shit. It's about being short and sweet and straight to the point, yeah. Cool. It's paragraph 3, yeah. Alright, safe. Now, again, on the right hand side, we see we got topic 5 in this year, new topic photosynthesis here. It's just gonna unlock more marks for us here because we're mentioning different shit here. And also topic 1 again, yeah. Alright, cool. Nice. Yeah. So what we need to talk about here, yeah, we wrote this down already, photosynthesis, yeah, LDR, all of that, yeah, ATP making, all of that, yeah. So this is another thing, yeah. I'm gonna teach you man a little cheat code, yeah. I'm gonna teach you man a little cheat code, yeah. Instead of learning bare extra information, yeah, what you can do, yeah, is you can make sure that you learn the processes, yeah, in extra information. So what I mean by that is photosynthesis, yeah. Look at like a video, yeah, of how to of like it in like some American video or something of it in like great detail, yeah, and memorize that, yeah, and it will help you a lot. It means that you got bare extra information just like that straight off a process, yeah. Because deep it, a lot of the essays, yeah, they're gonna revolve around processes, yeah, like photosynthesis, nutrient cycle, mass transport, yeah, um all that shit, respiration, yeah. So if you learn that shit in detail, yeah, so like photosynthesis in detail, yeah, you'll be able to pattern that easily. Do you see what I'm saying? Because you'll know all of this shit, which is at like a university standard, yeah, which means that that's an easy Gara paragraph, yeah, to get the extra marks, yeah. So this is the extra information paragraph that I included. This is it right here. So in plants, yeah, water is vital process of photosynthesis, especially during the light-dependent reaction that occur in thylakoid membranes of chloroplasts, yeah. So that's just straight A-level, yeah, that's standard, bro, yeah. Now, these reactions begin when light energy at certain wavelengths here. So when you start talking about wavelengths here, all these biology guys, they like that. You get me? All these teachers, they like that. When you start mixing physics with this shit here, they start liking that. You get me? So talk about wavelengths here. It's absorbed by photosynthetic pigments here and directed towards the reaction center of photosystem 2. Photosystem 2 is also called PS2, yeah. Don't be scared, yeah. That's a normal... You get me? Photosystem 2 is something normal, yeah. That's just what it's called, yeah. So you need to know that it's gonna occur in the reaction center of that, yeah. Again, you know all this information. If you look at photosynthesis just a little bit deeper, you'll know all of this, yeah. Specifically, the P680 special chlorophyll pair, yeah. So the actual chlorophyll pair that's in that photosystem, yeah, that the light actually absorbs onto, yeah, is called the P680 pair, yeah. So there's only one of them, it's the P680 one, yeah. So you including that, yeah. Imagine what the examiner's thinking now. He's thinking, damn, like this guy knows bears, yeah. Upon excitation, P680 enters an energetically unstable state, so obviously it's going to be in a higher energy state, yeah? If you man done physics, you know about electrons moving into a higher energy state, that kind of thing, photoelectric effect, all of that, yeah? And donates an electron to the primary electron acceptor, yeah? Phthalate, yeah? So phthalate is the primary electron acceptor. That's the molecule that accepts the electron at the start, yeah. So that's, again, you would know this if you just do a little bit more into photosynthesis, yeah. This electron transfer is known as a photoact, yeah. So that's when the first electron transfer, that direct excitation, yeah, we call that photoact, yeah. So photoact, and it initiates the electron transport chain, yeah. P680 is now positively charged and hence regains an electron through photolysis of water, yeah. So photolysis of water is inside A level, but you need to go a little bit more detail here, yeah. So this is catalyzed by a manganese calcium cluster, yeah. So this magnesium calcium cluster, yeah, it accepts certain things and it actually makes it's the reason it catalyzes the photolysis, yeah, of water, yeah. So that's what it actually comes down to. So you mentioning that manganese calcium cluster, again, that's that extra information that you know what you're talking about, yeah. This complex extracts 4 electrons from 2 water molecules. That's A level, but because we added that little complex thing, it's going to add a little bit more to it, you see. But you should know that it's 4 electrons with 2 water molecules, yeah, releasing 4 protons, yeah, into the thylakoid lumen, yeah, and 1 molecule of oxygen as a byproduct, which contributes to oxygen in the atmosphere. It's about these little things, bro. This is what separates you from like certain man, yeah. Adding these little things like contributes to oxygen in the atmosphere just makes your whole thing more concrete, yeah. And you got to think as well, structure each paragraph properly, bro. You got to think about how it's going to be read, yeah. Calm. And remember, you don't need everything here to get the full 25 marks. Yeah, this essay is very, very well done. Yeah, that's why it's gonna be like that. But you don't have to form it in this exact way. Of course, under pressure in the exam is different. Yeah. Calm. The electrons pass from phenolphthalein, yeah, to plastoquinine, yeah, through the cytochrome. So that's just the way that the electron transport chain is going through. Yeah, if you look at photosynthesis in a more detailed way, yeah, you'll see that it's actually steps for how the electron transport chain moves. Yeah, and you'll just memorize that it's in that order that it moves through. Yeah, so from this to that to that to that. Yeah. Gradually losing energy that is used to actively transport protons into the thylakoid lumen, yeah, creating a proton gradient, yeah. Calm. So that's that paragraph there done, yeah. That's the extra information paragraph patterned, yeah. That's gonna get you the 25 marks right there, yeah. Cool, yeah. This is paragraph 4, yeah. Again, so in the plan, yeah, I wrote down ATP and NADPH production, yeah. So that's part of photosynthesis, yeah, and you can go into this in a deeper way as well. So if you look at ATP and NADPH production, yeah, you can start talking about that, yeah. So this gradient, yeah, this gradient refers to the previous paragraph. We talk about the proton gradient, yeah. It's central to chemiosmosis, yeah, as it drives protons back into the stroma through ATP synthase, which resynthesize ATP. Okay, calm. That's A-level standard, yeah. That's calm, yeah. At the same time, the electron reaches photosystem 1, yeah, PS1, yeah. Alright, cool. So that one there, yeah, is not, that's extra a little bit, yeah. Where it re-energizes a P700 chlorophyll pair, yeah. Now, you're probably thinking, how can I know that, yeah? You just know it from now. So these little things, when you watch these kind of videos and you learn slowly, yeah, you'll start building up knowledge, you get me? So you see which one, the chlorophyll pair for photosynthesis LDR. Now you got the chlorophyll pair for NADP production, yeah, which passes through the electron to ferredoxin and then to NADP through NADP reductase, yeah. Now NADP reductase is gonna reduce NADP, yeah, to make NADPH, yeah. Both ATP and NADPH are essential for the Calvin cycle, enabling the fixation of carbon dioxide. So this is more A-level part, yeah. So I talked about a little extra information there, NADP reductase, yeah. And now you're going to add the little A-level part to back it up here. So you're going to talk about for the Calvin cycle, enabling fixation of carbon dioxide as the production of triose phosphate, a precursor to glucose and other organic molecules. That's obviously from respiration part of A-level, yeah. Without water, this process would not occur as electrons would not flow. ATP and NADPH would not form and carbon fixation would cease, yeah. Hence, water is not only a reactant in photolysis, yeah, but also the foundation of the entire photosynthetic process and consequently for the plant's ability to grow, store energy and contribute to global oxygen levels. Because we talked about the oxygen moving into the environment, you can add that part at the end there as well. And again, there's a very concrete paragraph here. Calm. Calm. This is paragraph 5, yeah. Cool. So again, we got mass transport, yeah. So now another topic here, topic 3, yeah. Topic 1 and then topic 1 again, yeah. Cool. So water's physical properties, yeah, also enable efficient transport within plants through the xylem, yeah. The cohesion tension theory explains how water molecules linked by hydrogen bonds, yeah, cohesion. So that's using the cohesion property, yeah, which is just a normal A-level, yeah. Are pulled upwards from the roots to the leaves as water evaporates from the stomata during transpiration. So you're mentioning transpiration stream as well, which is also A-level standard, yeah. adhesion between water molecules and the hydrophilic walls of xylem vessels, yeah. So instead of just talking about, so you're talking about adhesion, yeah, which is obviously A level, yeah. Instead of just saying that water is gonna move up here through the xylem, yeah, you're talking about the hydrophilic walls, yeah, which is obviously an A level kind of word, yeah. The thing with these essays, yeah, you know everything, like you know already, like what you need to write. You already know all the information. It's just about how to write it. You get me? Alright, cool. This further aids this upward movement, yeah. Before reaching the xylem, water moves through the soil, through the root, through the two main pathways, yes. Again, this is normal, bro, standard A level, yeah. Apoplast, why did I say it like that? Apoplast pathway, yeah, where water travels through cell walls and intercellular spaces, yeah. So that's without crossing cell membranes, yeah. So that's when it squeezes through the cell walls and just moves through, yeah. And then simplast pathway, yeah, is where the water moves through the cytoplasm of the cells, connected by the plasmo desmata, yeah, which are small channels that allow direct flow between cells, yeah. This combination of pathways ensures efficient movement of water and solutes to the xylem. This continuous column of water not only supplies necessary reactants for photosynthesis, but also facilitates the transport of dissolved minerals, yeah, and helps regulate leaf temperature through evaporative cooling, yeah. Maintaining optimal temperature is crucial for enzyme function and overall metabolic processes, yeah. Furthermore, water contributes to the cell turgor, yeah. So again, I wrote down in the plan to mention turgor as well, yeah, because I studied with water helping the structure of the cell, yeah, providing structural support to leafy parts of the cell, yeah, of the plant, yeah, and providing role in growth and nutrient transport, yeah. So this paragraph here is calm, it's easy. It's just using a small part of the A level course, yeah. There's no extra information in here at all, yeah. But obviously, it's about how to frame that. Do you see what I'm saying? Alright, calm. It's paragraph 6 of the essay, yeah? So it's just a little continuation of paragraph 5, yeah? So I'm going to talk about in the phloem, yeah, what is integral to the translocation of organic solutes, yeah? Such as sucrose from source cells to sink cells, yeah? This process relies on the pressure flow mechanism, yeah? So this is all just A level as well. We need to talk about like the flow, the the source to sink kind of theory, yeah, where active transport of sucrose into the CF tube elements here lowers the water potential. Notice how I'm not talking about the whole process. I'm not saying that's moving into the CF tube and then back and all that shit, yeah. I'm just saying, yeah, moves into the CF tube elements, yeah, to lower the water potential, causing water to enter by osmosis from adjacent xylem vessels. That's all you need to say. That's just a little quick summary. That's it, bro. The actual like knowledge you need is very small knowledge, bro. You don't need to have like that much stuff here, but you just need to put it in the correct order. You get me? The resultant hydrostatic pressure drives the flow of the phloem sap towards the sink cells, yeah, where the sucrose is unloaded obviously into the leaves, yeah, and water exits the phloem, returning to the xylem. This system ensures the distribution of essential nutrients throughout the plant, which will support growth and storage, yeah. So that's it, bro. Very short, very concise paragraph, bro. Just kind of solidifying the extra marks that it's gonna have there, yeah. Calm. You're probably thinking, yeah, how are you gonna fit all of this in an essay, yeah? That's the thing though. A lot of people get distracted about talking about specifics, bro. It's not about the specifics, bro. It's gotta be short and impactful, bro. You can't just have bare shit. You see what I'm saying? Certain people are gonna go into the like details of this, yeah. Talk about how the xylem is structured, all of that. It's not about that, bro. It's about how the water, specifically the water, yeah, is helping the xylem. You get me? It's about how the water is important, yeah. That's it. Cool. It's paragraph 7 or whatever. Yeah. Alright. Cool. Safe. So here I'm gonna talk about topic 6 here. So I'm gonna bring topic 6 into that to get more marks here. Remember, you need 3 different parts of the course here. Alright. Calm. So here I'm gonna talk about ADH and control of water potential, blood water potential, osmoregulation, all of that. Yeah. So osmoregulation is vital for maintaining cellular homeostasis. Yeah. Calm. Osmoreceptors in the hypothalamus detect changes in blood water potential, yeah. That's just normal A level, yeah. Prompting the release of ADH, yeah, from the pituitary gland, yeah. ADH increases the permeability of the distal convoluted tubes, yeah, and collecting ducts, yeah, in the nephrons, yeah, of the kidneys by stimulating the insertion of aquaporin channels into their membranes, yeah. So notice how for every single chapter, yeah, every single paragraph here, it's mainly just A level shit. Apart from the second one about the photosynthesis here, it's mainly A level shit. But like I add like 1 or 2 words, I add something extra, something there, yeah, like the aquaporin, yeah. This allows more water to be reabsorbed into the bloodstream, concentrating the urine and restoring blood water potential, yeah. Proper osmoregulation prevents cell from undergoing lysis due to excessive water intake or crenation due to dehydration, ensuring that cellular processes are efficient, yeah, at comms. That's all you need to say here, yeah. Just topic 6, standard, bro. Again, a little summary of it, bro. It's not like in detail, yeah. It's just summary, bro. Just talking about how hypothalamus is detecting the change, and then talk about the ADH, yeah, increasing the permeability of the distal convoluted tubes, yeah, and the collecting ducts, yeah, and all of that, and then just talk about how that leads to whatnot, yeah. Alright, cool. Alright, cool. So Paragraph 8, yeah, I talk about tissue fluid, yeah, which is in topic 3. Yeah. If you go look at topic 3, you'll see I talk about tissue fluid and how it's formed and everything in detail, yeah. Alright, calm. So, water also plays a pivotal role, yeah, in the formation of reabsorption of tissue fluid, yeah, which bathes cells and facilitates the exchange of gases, yeah, nutrients and waste products, yeah. At the arterial end of capillaries, high hydrostatic pressure forces plasma out of the um interstitial spaces, yeah, forming tissue fluid. At the venous end, yeah, so at the venous end, it's always the lower hydrostatic pressure, yeah, and higher osmotic pressure, yeah, due to plasma proteins like albumin, yeah. So this albumin shit, yeah, it's just normal proteins inside the plasma that obviously makes um the water potential lower, yeah. But listen, yeah, the whole point of this, yeah, what I'm trying to say is that again, it's a normal A-level paragraph, yeah. You should be able to write this, yeah. But the only extra part is that albumin part. You see what I'm saying? So it still keeps it at that kind of standard, bro. You get me? And obviously that draws water back into the capillaries via osmosis because obviously you're lowering the water potential, yeah. This dynamic equilibrium maintains tissue fluid balance, ensuring that the cells remain hydrated and metabolic waste is efficiently removed. Now again, you're going to add a little bit more extra information here. Disruption of this balance can lead to edema, yeah, impaired nutrient delivery and accumulation of toxic substances adversely affecting cellular function. So that edema like thing is just a little extra thing, yeah. So what I'm saying, yeah, is from what I ever mentioned inside the PowerPoints, yeah, just take that as that extra information that you can sprinkle into the PowerPoints that have the A-level standard, yeah, and then also memorize the actual processes like photosynthesis and all of that in detail, yeah, so that you can make that into a big paragraph on its own and you can copy paste for any essay, yeah. Alright, calm. Alright, cool. So paragraph 9, yeah. So as you see, inside the previous inside the plan, yeah, I was talking about gills, yeah, and like fish and all that shit, yeah. So he want to talk about that as well. So in fish, yeah, water is a medium for, of gas exchange. Yeah, fish have gills with filaments and lamellae that provide a large surface area for diffusion. That's just A-level calm, yeah. Now A-level, you talk about the countercurrent flow mechanism, yeah. The countercurrent flow mechanism, where water and blood flow in opposite directions across the gill surfaces, maintains a concentration gradient that maximizes oxygen uptake and carbon dioxide removal, yeah. The efficient gas exchange system supports aerobic respiration, where oxygen acts as a terminal electron acceptor, yeah. So again, we're mixing a bit of the aerobic respiration into that, yeah, which is both topic 5, so 5, so it's calm, yeah. But yeah, alright, calm. And electron transpiration, yeah, enabling the production of ATP, yeah. Without adequate water flow and oxygen availability, fish would experience hypoxia, yeah, leading to reduced energy production and death. Yeah, so obviously, again, not too much information here, yeah. We just broke it down into the gill filaments, the lamellae, yeah, the counter current flow, high oxygen uptake. But you can imagine your teacher just like highlighting the shit. Imagine the examiner just highlighting like he's, you know what I'm saying? You give it what you need to give it. You get what I'm saying? And you also sprinkle a little bit of the aerobic respiration in there as well. You know what I'm saying? Alright, calm. But because you mentioned that, it brings from other part of the course here. My man, he knows the whole course in it, the examiner. So he's gonna be writing these kind of codes here next to it from the specification. Yeah, you get it? Alright, cool. During sweating, yeah, water absorbs a significant amounts of heat from the body as it evaporates from the skin, effectively cooling the body and preventing hypothermia, yeah. This mechanism is essential for maintaining enzyme activity and overall metabolism, yeah, specifically in endothermic reactions that rely on internal temperature regulation, yeah. So just nice little bridge again, yeah, just nice little thing to lock it in, you know what I'm saying, bro? Nothing too deep here. Alright, alright, cool. And now, final paragraph here, paragraph 11 here. You just want to wrap it up with a nice little, you know what I'm saying, outro, yeah, if you got the time for that, yeah. So, in conclusion, water's chemical and physical properties, yeah, make it essential for an array of biological processes, yeah. From facilitating metabolic reactions, yeah, and enabling nutrient transport to regulating temperature and maintaining cellular homeostasis, yeah. Water is integral to survive and functioning of all living organisms, yeah. Living organisms, yeah. Alright, calm. Alright, safe. So that's all of that, yeah. All of that completely patterned there, you know what I'm saying, bro. All of the essay there, yeah. Listen, fam, in the future, what I'll do, yeah, is I'll talk about the stages of the essay. I'll give you an example of like a low tier essay, yeah. And I'll talk about how that can be improved into a mid tier one and how can that be improved into another one and that, you know what I'm saying? So you can kind of build up to get to this kind of stage here, yeah. Now, you don't have to take every single paragraph here, of course, yeah. This is 11 paragraphs. Obviously, this has been highly like looked into, yeah, highly used the internet, that kind of thing, yeah. But this is just like a little idea, bro. You get what I'm saying? Little taste of what's to come. Do you know what I'm saying? So you just gotta kind of like pattern that, and eventually you'll be calm. Yeah. Alright, calm. So that's a perfect essay right there. Yeah, 25 marks guaranteed, bro. This is more than 25 marks, bro, because of the way it's structured here. But there you go, bro. Innit? Alright, calm, fam. Alright, cool. This is the inorganic ions sheet. Yeah. For inorganic ions, the first question is the essay. So it says the importance of ions and metabolic processes. Yeah. So you need to do what we always do. Yeah, which is write the plan. Yeah. So for the plan, you just want to think of any little any like topics you can think of here. So just say like, for example, I can say action potentials here and resting potential here. So I can talk about the role of the sodium potassium ATP hydrolase pump here. Alright, cool. That's one topic here. Another topic I can think of with ions here is going to be co-transport here. So co-transport in epithelium and kidney here. What I mean by kidney is I can talk about in the proximal convoluted tube here, like topic 6 and that here. Alright, cool. So I can say like um co-transport of amino acids here and carbohydrates and glucose. I can say as well that function in selective reabsorption here and proximal convoluted tube here. Alright, cool. Alright, calm, yeah. Also, I can talk about muscle contraction, yeah. So things like calcium ions being used and troponin and tropomyosin, yeah. Another thing I can talk about is like pH regulation, so like homeostasis and that, yeah. pH regulation to make sure that the blood pH is the same and that kind of thing. I can also put a little bit of extra things in there as well, like talk about the carbonic acid buffer system, yeah. Obviously, pH is based on hydrogen concentration, yeah. So I can talk about how hydrogen concentration is regulated in the blood, yeah. Alright, cool. I can also talk about things like iron and hemoglobin. I can talk about the Bohr effect with carbon dioxide, yeah, in the hemoglobin, as well as also talking about like electron transport chain, yeah. So obviously iron's used in that as well. Alright, cool. What else can I talk about? Yeah, I can talk about as well. I can talk about phosphate ions, yeah, in oxidative phosphorylation to make ATP, yeah, and also in DNA and RNA. Alright, cool. And then I can also talk about electrophoresis here and topic 8 and that. So talk about DNA genetic technology here. I can talk about like electrophoresis, that kind of thing, yeah. And also I can talk about chlorine shift as well. That's extra information, yeah. But always you should have like 4 different paragraphs here, 3 paragraphs from across the different specifications here, and then 1 paragraph of like extra information. You get me? So that's gonna be the extra information one, yeah. So now we're gonna get into the essay here. Alright, calm. So this is the first paragraph I'm gonna go through, yeah. So um this paragraph here is gonna include topic 2 here and topic 6 here. So already you got 2 topics locked in the bag here. You get me? You got topic 6 like nervous coordination and that. You also got topic 2 as well, talking about transport across membranes. Yeah. Calm. So what I can say is that the resting membrane potential is -70. Yeah. So that's just the resting potential. That's straight A-level knowledge. Yeah. In neurons. This is due to the asymmetrical distribution of sodium plus and potassium plus ions across a plasma membrane, maintained by sodium potassium ATP hydrolase pump here. So you can just say sodium potassium pump here, but I like to add a little bit of, you know what I'm saying, flavor to it, yeah? So that's why I said ATP hydrolase pump here, because it's going to be running on ATP hydrolysis, obviously, yeah? This pump actively transports free sodium out of the axon and 2 into the axon, yeah? Per ATP molecule hydrolyzed, yeah? So that's just a little bit of information there. That's straight from the A-level course, not really anything added there, yeah? This creates both concentration gradients and an electrical gradient inside that is relatively less positive compared to the outside the axon, yeah? So obviously there's less positive ions inside, so therefore the inside is less positive compared to the outside, yeah? That's going to create not only a concentration gradient, which is going to be needed later for the sodium to move in due to the influx, yeah? But it's also going to make the electrical gradient needed for the depolarization to occur, yeah? When a stimulus reaches the threshold potential of negative 55 millivolts here, voltage-gated sodium channels open, allowing rapid sodium influx, depolarizing the membrane to positive 40 millivolts here. So obviously, when it reaches the all or nothing principle here, negative 55, it's going to make all the sodium channels open, and it's going to move all the sodium into the axon here, making it positive here. That's obviously depolarization, yeah. So this followed by the opening of voltage-gated potassium channels causing potassium efflux here. So that means that potassium is now leaving the axon here. There's bare potassium leaving here that repolarizes the membrane. A hyperpolarization phase occurs due to the excess potassium leaving as potassium channels take a while to close, briefly making the membrane more negative. Refractory periods ensure unidirectional action potential propagation. These electrical signals are the foundation of rapid communication in the nervous system here. So that should lock in a lot of marks there, yeah. That's just straight from A-level course, nothing added, no extra information, yeah. And you're talking about as well at the end why it's important, why the ions are important. You're saying that it's gonna cause electrical signals are the foundation of rapid communication in the nervous system, yeah. Alright, cool. Alright, cool. This is paragraph 2, yeah. So sodium ions are essential in secondary active transport mechanisms across epithelial cells in both the small intestine and proximal convoluted tube of the nephron, yeah. The sodium potassium pump on the basolateral membrane, so that means that the side that's facing the blood here, establishes a low intracellular sodium concentration here. So what that's trying to say is that it's facing the blood here, it's gonna push sodium outside, yeah. So extracellular, yeah, is gonna have a high sodium concentration so it can move back in and move glucose with it, yeah. You get me? This gradient allows sodium to diffuse in through simple co-transporters, yeah, coupled with either glucose through SGLT1, yeah. So that's a little bit of extra information, yeah. SGLT1 is a very common protein, yeah. All it is, it stands for sodium glucose link transporter 1, yeah. And that's just inside the ileum, and it's just gonna bring the glucose with myman into the cell, yeah. And that's called secondary active transport because it's gonna use energy from the sodium gradient instead of ATP, yeah. So it's from AEP, but it's mainly from the sodium that's moving in. You get me? Um, or amino acids, yeah. Alright, cool. The solutes then passively diffuse into the blood through facilitated diffusion through the GLUT transporters, yeah, such as GLUT2, yeah. So GLUT is a similar thing, yeah. So these are the ones that make glucose move out of the cell into the blood, yeah. So it's basically gonna be there to move it into the actual blood itself, yeah. So getting the glucose directly into the blood, yeah. And that's gonna be through facilitated diffusion, yeah. So in the proximal convoluted tube, similar sodium-dependent co-transport mechanisms reclaim essential nutrients from the glomerular filtrate with which aids selective reabsorption. Yeah, so obviously the filtrate that's moving through the nephron, yeah, through the glomerulus first, yeah, that's gonna be obviously used to co-transport to reclaim any essential nutrients that's inside of it, yeah, that needs to go back into the blood, yeah. So that's gonna touch on topic 2, topic 3, and also topic 6 here, because you're talking about digestion and shit. You're talking about like transport, topic 2, and you're also talking about like control of blood water potential, all of that. That's topic 6. Alright, calm. Cool. This is paragraph 3. Yeah, I'm gonna talk about all of that. So calcium ions play a central role in excitation and contraction of muscle fibers. Upon arrival at a nerve impulse at a neuromuscular junction here, acetylcholine is released here. So that's just normal topic 6. You get me? Straight A-level content here, which binds to receptors in the sarcomere, triggering depolarization and the propagation of action potential along T tubules here. T tubules is a little bit of extra information. All that is that's the actual like tubes that conduct the action potential, yeah, because they're gonna move from like deep inside the muscle fibers to the junction, yeah. So through that is gonna be through the T tubes, yeah. This leads to the opening of voltage sensitive calcium channels in the sarcoplasmic reticulum, releasing calcium into the sarcoplasm, yeah. So obviously the sarcoplasmic reticulum, yeah, is what contains all the calcium ions, and then when the action potential reaches it, yeah, it's gonna release all of that into the actual sarcomere, yeah. Calcium binds to troponin, yeah, inducing a conformational shift in troponin tropomyosin complex, yeah. So obviously, that's the like the safety cover around the muscle, yeah. That's gonna prevent it from sticking. When it shifts, that creates a conformational shift, yeah. That's gonna expose it into the actin, into the myosin binding sites here on the actin filaments, yeah. Then the cross bridge is gonna form, which is powered by ATP hydrolysis, and then calcium is actively pumped back into the sarcoplasmic reticulum after contraction using calcium ATP hydrolase, yeah, allowing the muscle to relax. Alright, cool. So that's just mainly using topic 6 and topic 1, yeah. So talking about skeletal muscles, talking about nervous coordination, that kind of thing, and ATP, yeah. Alright, calm. Alright, hydrogen ions, yeah, determine pH, influence in enzyme activity, protein conformation, and metabolic rates. Enzymes have optimum pH values, and deviation leads to altered ionic interactions and hydrogen bonding, disturbing tertiary and quaternary structures here. So obviously that's what makes it A-level here, by talking about tertiary and quaternary structures here. So in the tertiary structure, you got ionic interactions here, and you also got hydrogen bonding as well. So obviously having different hydrogen ions here is gonna make the pH different, maybe more acidic, that kind of thing here. That's gonna denature it by altering the hydrogen bonds and shit like that, interfering with it here. And then you got the carbonic acid buffer system here, which is obviously a little bit of extra information here, but basically the blood's got to maintain its pH level here, and by doing that, pretty much here, the carbonic acid inside the blood here, which is formed from obviously carbon dioxide dissolving into it here, splits, because it's a weak acid here, it's going to split into H plus and HCO3 minus here, and that's going to maintain blood pH at 7.4 here. You should know that here, that blood pH is around 7.4 here. In tissue, CO2 combines with water catalyzed by carbonic anhydrase, producing H+, which may bind to hemoglobin, causing the haldane effect here. Haldane effect here is just like the H+ is going to bind to hemoglobin here, which is obviously going to reduce the H+ concentration in the blood, which is going to make it less acidic, you get me? Because obviously you've got more H+, that's going to make it more acidic here. It's like chemistry here. Don't get confused by these things, bro. Carbonic anhydrase, all of that, it's just obviously HCO3 minus here without the extra H here. That's all it is, bro. It's just normal chemistry, fam, yeah? Alright, cool. Alright, cool. Iron is essential in the heme phosphatic group here of hemoglobin. Each Fe2 ion here in the center of the heme group can reversibly bind into one oxygen molecule, enabling oxygen loading in alveolar capillaries where partial pressure of O2 is high, and unloading in tissues where partial pressure of O2 is low, yeah? So that's just standard A level, you get me? Standard normal shit, yeah? Topic 3 about like partial pressure of oxygen, that kind of thing, yeah? Then you could say that cooperative binding nature of hemoglobin results in the sigmoid shape of the oxygen dissociation curve here. So that's again just talking about the dissociation curve, talking about why it increases steadily due to cooperative binding, which obviously due to the iron phosphatic group here, which is obviously an ion here. Factors like increased CO2, that's going to be Bohr effect here, shift the curve to the right, facilitating oxygen unloading during exercise or metabolic stress here. Iron also plays a role in cytochromes in the electron transport chain, cycling between Fe2 plus and Fe3 plus here. My mouse tweaking, fam. As they transfer electrons here. So obviously what that means here is just that basically the cytochrome is just like a heme containing group here. Obviously the electron needs to move through the transport chain, and that's going to be through redox, you get me? So it's going to be Fe2, then it's going to donate an electron, become Fe3 plus, like that, and alternates, you get me? Alright, cool. Alright, cool. So now what you want to do is you want to talk about how oxidative phosphorylation leads to ATP production, yeah, which is obviously going to be needed as energy for the whole body. So you need the ions of phosphate, yeah. So I can say that phosphate ions play a vital role in metabolic processes, especially in energy transfer and synthesis here. In oxidative phosphorylation, which occurs in a mitochondrial membrane, reduced coenzymes NADH and FADH2 donate electrons to the electron transport chain. As electrons pass along a series of electron carriers, energy is released and used to actively transport hydrogen ions here from the mitochondrial matrix to the intermembrane space here. This creates an electrochemical gradient, also known as the proton motive force here. H plus ions then diffuse back into the matrix through ATP synthase, driving the phosphorylation of ADP and inorganic phosphate to form ATP here. So that's just standard A-level biology here. This process is known as chemiosmosis and is the method in which ATP is formed here. The final electron acceptor is oxygen, which combines with electrons and H plus to form water here. The phosphate ion is not only involved in ATP formation, but also in transferring energy within the cell. Once formed, ATP can phosphorylate other molecules using its inorganic phosphate group, making them more reactive. This is essential for processes such as glycolysis here, and glycolysis is needed to phosphorylate here, where glucose is phosphorylated to allow further breakdown. Therefore, phosphate ions are fundamental to both production and use of energy in cells. Another major function is the structure of DNA and RNA. Phosphate ions form part of the phosphate backbone by linking sugar molecules of nucleotides together through the phosphodiester bonds here. These bonds are essential for the stability and structure of nucleic acids here. So this paragraph is just a solid, like, fully A-level paragraph here. There's no extra information here, yeah? It's just straight A-level here. It touches on topic 1. It also touches on topic 5 properly for the first time in the essay, yeah? Which means that you're also going to expand your marks into there, yeah? And also is going to be touching on topic 4 as well with DNA, like synthesis, that kind of thing, yeah? Alright, cool. Alright, cool. Now this paragraph here, you want to bring topic 8 into it, yeah? To really encompass that, like, marks here, get all the marks here patterned, yeah? So what you want to say here is that electrophoresis is a core technique in molecular genetics for separating macromolecules, especially DNA fragments, yeah? So that's talking about topic 8, yeah? by size and charge through an agarose gel under an electric field, yeah? So that's just normal A-level, nothing extra there, yeah? The buffer solution made of TAE, so that's going to be a little bit extra, yeah? So again, you can go look at extra information. I'll make more about extra information in the future. It will have these kind of things here. So TAE is tris acetate EDTA, yeah? You should know a little bit about EDTA in the chemistry if you do A-level chemistry. Or TBE, yeah, tris borate EDTA, and it contains inorganic ions such as tris plus, yeah. So that's where you're bringing the extra information about inorganic ions into it, yeah, because inorganic ions are obviously that's the whole point of this essay, yeah, is the importance of that, yeah, which maintains pH stability, yeah. So the reason why you have that tris plus ion, yeah, like TAE, yeah, is because that's gonna basically stop the DNA from getting destroyed and stop denaturization, yeah, of anything inside it when you're doing this electrophoresis, yeah, to separate the DNA and to make it obviously, you know what I'm saying, do what it's meant to do, yeah. Obviously, the DNA is gonna travel through the gel, yeah. Calm, which maintains pH stability, yeah. Acetate and borate minus, yeah, which provides ionic conductivity, yeah. So that's the reason why it travels through the acetate and borate, yeah. They're gonna make that ionic conductivity and that electrical like charge that it can flow through that, yeah. And EDTA, which traps cations like Mg2 plus here to inhibit DNA's activity. Yeah, so DNase is gonna break down DNA. So obviously, you don't want it to break down the DNA when the DNA is exposed like that. Yeah, so what you can do is you use EDTA. Yeah, it's gonna trap things like Mg2 plus. When it traps Mg2 plus here, it stops DNA's activity. Basically, it stops DNA enzymes from working. Yeah, preserving DNA integrity. Yeah, DNA being negatively charged due to its phosphate backbone migrates towards the anode. Yeah, so that's normal, bro. The rate of migration depends on molecular weight and agarose concentration. Yeah, so obviously it depends on the agarose concentration inside the gel. It's also going to depend on like how much it weighs. Obviously, yeah, the lighter one's going to move faster. Yeah, smaller fragments migrate faster through the porous gel. The precise function of ions is in the buffer is to facilitate consistent current flow, prevent overheating, and stabilize pH, ensuring reproducible and accurate results in applications such as genetic fingerprinting. Yeah, restriction mapping and PCR verification. Yeah. So that's a little bit of extra information put into that. And with that paragraph here, you're basically pattern, bro. That's like full 25 out of 25, yeah. Perfect, yeah. You got like more than three topics here. But I'm gonna add another little paragraph as well of extra information, just for like your own knowledge as well, so you can use it in the future, yeah. Alright, calm. Alright, cool. Alright, safe. Last paragraph, yeah. So you don't really need to know about this, but it's just the extra little information. You can use that in the future whenever you need to use it, yeah. Alright, in red blood cells, CO minus is integral to chloride shift here. So chloride shift is basically, I talked about the little buffer effect with the CO2 and the H plus and the carbonic acid, all of that, yeah, to make sure that the pH is remaining the same. This HCO3 is gonna move away, yeah, and when it moves away, because it's negatively charged, yeah, that's gonna like lose negative charge. So you need to make up for that loss of negative charge, yeah, to make it electrochemical neutrality, yeah. So what you need is like another like CO minus, another negatively charged particle, yeah, to move in, yeah. So that's why the CO minus moves in. That's called a chloride shift, yeah. Alright, cool. This maintains electrochemical neutrality during CO2 transport, yeah, from tissues to the lungs, yeah. This ion exchange is mediated by the anion exchanger protein, yeah, AE1, yeah. So you just need to know that. That's like a little, you know what I'm saying? AE1 is basically the protein that basically mediates that, yeah, and ensures efficient CO2 buffering, yeah. In the nervous system, CO minus is pivotal in generating postsynaptic potentials, yeah, that inhibit it, yeah, through GABA A receptor, yeah, activation. So GABA A receptor is what GABA binds to, yeah. So when GABA binds to these ligand gated ion channels, so ligand just means that it's GABA is a ligand, yeah. So it's just that shit that binds to it. You get me? You can look at it more depth in chemistry. Yeah, alright, cool. CO minus influx hyperpolarizes the postsynaptic membrane, reducing the likelihood of action potential firing, and helps keeping the nervous system under control. Yeah, so certain things here can cause GABA to release here and attach to the GABA A receptor, which is basically just gonna make like it's basically just gonna stop the action potential from moving along. Yeah, things like Xanax and shit like that it can bind to it. It can cause them kind of things. Yeah, alright, safe. Alright, cool. Yeah, question 2A. So basically, yeah, it's saying that you got aspirin, yeah, and you got paracetamol, yeah, and it's asking which one, like, if he takes the maximum of both of them, yeah, is it gonna be over the recommended sodium allowance, yeah? So for aspirin, yeah, so just put aspirin, yeah, and one tablet is 0.3 grams, yeah. So hence, it's question A, by the way. So hence, yeah, how much aspirin, yeah, is the maximum, yeah? So the max is equal to 0.6 every 4 hours, yeah, in 24 hours, which is how many hours there is in the typical day, yeah. that's basically going to be 6 times, yeah. So 6 times 4 is 24, yeah. So basically, you need to times the 0.6 every 4 hours, yeah. So that means 0.6 times 6, yeah. Because 6 times 4 is how many hours in a day, yeah. That's going to be 3.6. And now you got 3.6 grams maximum in a day, yeah. So how many tablets is that, yeah? Well, it's 0.3 grams in one tablet, so 3.6 divided by 0.3 is going to be 12, yeah? So that means it's 12 tablets, yeah? So 12 tablets of aspirin, yeah? And then what's the mass of sodium in a tablet, yeah? 0.15, so 12 times 0.15 is going to be 1.8. So is that above the recommended daily allowance? Yeah, it's not, because the recommended daily allowance is 2.4, yeah? So hence it's no for aspirin, yeah? Now you need to do the same shit for paracetamol, yeah? So just put paracetamol, calm. One tab of paracetamol is zero point 0.5 according to the table, because it's 500 milligrams here. Calm. Alright, safe. So that's one tab here. And how many? So it's every 6 hours, yeah. So how many? 6 times 4 is 24. So it's basically gonna be 4 times, yeah. And so 1 gram every 4 times. So 1 times 4 is gonna be 4 grams in total, yeah. So it's 4 grams here, and in one tablet there's 0.5. So you need to do 4 grams per tablet per 0.5 to find out how many tablets you took here. He's gonna take 8 tablets here. So he took 8 tablets here. So now just times 8 by the sodium in one tablet. So 8 times 0.43 from the table. That's going to be 3.44, yeah. So therefore, yes, it is above the daily recommended value for that, yeah, for paracetamol, yeah. Alright, calm. Alright, question B. It's just asking about what factors should you keep the same in experiments or any like clinical trial kind of experimentations, yeah. It's always going to be things like gender, age, shit like that, ethnicity, them kind of things, yeah. So just put age and gender, yeah. Alright, cool. And the thing that's different, yeah, is obviously the concentration of sodium in the medicine, yeah. Trying to see if medicine has any effect here. So, sodium has any effect here. So one of them is gonna have sodium. One's not gonna have sodium. Yeah. So concentration of sodium is the changing factor. Yeah. Alright. Cool. Question C. Calculate how many of the control patients had hypertension. Yeah. So basically, yeah, 4.73% had taken a medication. Yeah. So 4.73. Yeah. So 4.73%. Yeah. So that's over 100. Yeah. Times that by the total number of patients. Yeah. So 1, 2. 1 million, yeah, 292,000. That's in the line of information above, yeah, before the part B. 337, yeah. That's gonna be 61127.54, yeah. And then it says that it's gonna be 7.18 times fewer, yeah, control patients with hypertension. So divide that number by 7.18, yeah. So 61127.54. Divide that number by 7.18, yeah. And you're gonna get how many with hypertension. So 8513.5. Obviously this is people, yeah, so you can't have like zero point 0.5, so you just put 8513. It's always better to do the lower number, yeah, but it doesn't really matter. You can put 8514 as well, you're still gonna mark here. Alright, cool. D. Alright, so D is basically asking, yeah, explain how a high concentration of sodium is gonna lead to hypertension, yeah. So obviously the reason for that is because sodium ions are gonna lower the water potential of the blood, yeah. So the sodium ions. You're gonna increase blood volume, yeah. So basically, yeah, sodium ions here. So when you got more concentrated shit like ions and that in the blood, yeah, it's gonna lower the water potential, yeah, because you got less room for water, yeah. So therefore, water's gonna need to move in to fill that up, yeah. So from higher to lower, yeah, by osmosis, yeah, which is obviously gonna increase the blood volume. So therefore, you get me, standard, bro, yeah. Alright, cool. Question 3, yeah. Describe the roles of iron ions, yeah? Sodium ions and phosphate ions in cells, yeah? So just basically state which one is which, yeah? So say that iron ions, yeah? To bind with oxygen, yeah? Sodium ions, you can say. You just need a maximum of 5 marks, yeah? So obviously sodium potassium pump moves out the sodium, yeah, and it's gonna come back in with the glucose, yeah, for the co-transporter protein, yeah? Alright, cool. You can also say that it creates a sodium concentration gradient, yeah? That's unrelated. You can put for both of them, yeah? So you can put for that, yeah, but you could also say like in other parts like the nephron, that kind of thing, yeah? So sodium concentration gradient's always there, yeah? Alright, cool. And then phosphate ions, yeah? Joins nucleotides, yeah? To form phosphodiester bonds, yeah? In the DNA backbone, yeah? And also used to produce ATP. And finally, I can say that's also phosphorylates the compounds, making them more reactive, yeah? So that's just straight topic one, easy, yeah? Alright, calm. That's it, bro, yeah? Listen, obviously topic one's a big chapter, yeah? Big topic, but you get me. It's minor, bro, yeah? Over time, it'll be pattern, man. Minor.